You're listening to the Common Descent Podcast.
Hello, Will.
Hello, David.
Hello, everybody, and welcome to Common Descent, where we geek out about paleontology and Earth history and stuff. This is episode 201, the second part of our grand celebratory trilogy in honor of episode 200. We are talking about the most requested topic in the history of the podcast, fungi.
Still.
Last episode was fungi. This episode, because it takes more than one episode to go over an entire kingdom and the thing that every one of our listeners wanted us to talk about, this episode is fungi and plants.
A classic duo.
Yeah, the name a more iconic duo. Fungi and plants it's getting its own episode just for itself. Massive topic. We're going to talk about their interactions. We're going to talk about symbiotic relationships, parasitic relationships, and we'll go into the fossil record and explore how important the relationships between fungi and plants have been to the evolution of both groups.
This is a topic I've been waiting for because way back when we did symbiosis, I remember feeling bad for how little I said about this.
Well, now we got a whole episode. Also, to add to the celebratory excitement, despite the number in the title, this is a plants episode.
It is!
And there's only one way to do a plants episode, and that means that after the news, we will be joined by special fungus celebration guest, Dr. Allie Baumgartner.
Yeah, we always outsource these.
We always outsource the plant discussions. Allie's joining us for the middle part of our trilogy on fungi. Very exciting stuff.
Rejoice, Allie fans.
Now, last episode, we not only mentioned that fungi was the most requested episode topic in the podcast history, we also listed off all of the names of the people who requested it. Incidentally, separate from requests for fungi generally, the topic of fungus and plant relationships was also requested many times. This episode topic specifically was requested by Amber, Anthony, Artist on a Mountain, Brian, Derek, Franz, Luke, Trey, Zek, Wait, Cheryl, Fern, Hannah, Karen, Kyla, Raymond, Susanna, Josiah, and Tater Boy. For those keeping track, that was 19 people, which means that this is the fifth most requested episode. So fungi has made it into the top 5 twice.
Yep.
Thank you to everybody who requested this topic, to everybody who engages with us and supports us and has helped to get us all the way to 200 episodes and beyond.
So awesome.
It's fantastic. Hey, here's a few announcements. Number 1, we have a Patreon. The Patreon supports everything we do with the podcast. We are in a very exciting transitional period of the podcast. We recently became an official business. We've got plans for future stuff to do, new things we want to try out, new people we want to collaborate with, and all of that is possible because of the support that we receive from our supporters on Patreon, who allow us to pay for everything we need to pay for to keep this going and keep this growing, indeed. If you support us on Patreon, there's all sorts of cool benefits that you can get. We do bonus noise, which is extra news and just chit-chat between the two of us. We do live streams every month where our patrons can join and interact directly with us. Higher tier patrons get physical goodies like pins and shirts and stuff. And at a particular level, you get your name shouted out on the podcast. This episode, we would like to welcome new patrons, Hannah and Marcy.
Welcome. Thank you so much for your support.
Welcome and thank you, dear listeners. If you would like to lend us your support as well, you can find the link to Patreon down in the episode description. Our subscription costs are pretty low, and a little bit goes a long way for us.
Absolutely.
We also have some other stuff going on all throughout the year. We have been releasing episodes of Spotlight 2024. Those come out at the beginning of the month. So there's a new one coming out very shortly after this episode launches. Also, very shortly after this episode launches is the month of October.
Spooky.
And that's spooky season, which means this is actually the last episode before spooky officially starts this year.
Yep.
Each Saturday in October, we will be picking a monster from popular culture, popular fiction, and discussing how it might evolve in the real world. Spooky is always a ton of fun. This year's theme is tiny monsters.
Itty bitty monsters.
We've got a great lineup for this year's Spooky. It's been a ton of fun to put it together.
Yep.
And then in November, there will be a bonus Cute-E episode, as is now tradition, and a Spooky live stream.
Yes.
So listen in, join us, get on the Discord server and interact with people, chat about Spooky. We've got a whole channel just for talking about Spooky. We've got another channel specifically for speculative evolution. It's a great time to get into all of this exciting, geeky stuff.
Absolutely. So excited.
Also, speaking of bonus content, we recently put out another Patreon mini-episode compilation.
Yeah.
We do, one of the other things you can get on Patreon is your own personalized mini-episode at higher tiers. And every now and then we put a bunch of those into a little batch and we release them so everybody can enjoy them. The 5th one is out now, so check that out.
Man, 5. It's so cool that we've done that many.
Yeah, our patrons are the best.
They're so great.
Our audience is the best. Hey,
You all
audience,
rock.
you're the best. And one more thing, we want to give a shout out to another podcast called, fittingly for this episode, Flora Funga Podcast, whose host KK is someone we became acquainted with recently and who sent us over a little promo to share. KK is the host of Flora Funga Podcast, as well as a plant biologist looking to bridge the gap between plants, fungi, and humans. KK has a thirst for mycology, the study of fungi, and would love to educate the importance of plant and fungi interactions. Mushrooms and plants can and will help save the planet one hyphae at a time. Mushrooms can help break up plastics and radiation. Plants can communicate above and below ground with other plants and fungi. Without their connections, we would not be able to grow food for the population nor have flowers around the world. Did you know that the smell of fresh-cut grass is actually the plant yelling for help? Or that plants have some of the same disease genes as us? If you like to get down and nerdy with plants, fungi, and other science facts, find KK on the podcast Flora Funga Podcast. And we'll put a link down in the episode description so you can go check it out. Let's move on to the news. Before we get into our main topic every episode, we like to tackle some news from the science that intrigues us and interests us. Will, tell us about what's new.
I have news about an oddly toothy bird.
Okay.
Yeah. This is a fossil bird with teeth and new specimens that give us a different look at what it might have been eating from what we expected it would be eating.
Ooh.
This is research by Jingmai O'Connor et al. In Current Biology, and the article is by Paul Smaglick in Discover Magazine.
Find that link to that article in the blog post.
This is a study on a new enantiornith bird, or not a new, but new specimens of this bird.
Cool.
These were the Cretaceous ancient group of birds, separate from the lineage that leads to our modern birds, but they were the big popular birds back then that were all over the place and doing very well. Very similar in many ways to our birds, but also distinct. This study was looking at one in particular. This is Longipteryx and is from the early Cretaceous, found in northeastern China. So it dates back to about 120 million years old. A small bird, blue jay sized, so bird sized. It has a long beak, long rostrum, with teeth at the end, because enantiorniths had teeth at times.
Yes. It was a very popular thing in the Cretaceous for birds to have teeth.
Yes. This bird has teeth at the end of the beak, notably, and enlarged teeth.
Okay.
Larger teeth at the end of the beak. They were described as large, recurved, and heavily enameled.
Okay.
So, beefy teeth.
Yeah, and curved backwards.
Mm-hmm.
These sound like grabbing at prey kind of
exactly.
teeth.
They sound like predatory teeth, and that's very much how they were interpreted.
Yeah, well, kind of like crocs and other long-snouted predators will sometimes have those extra big curvy teeth at the front.
Toward the tip of the snout.
Yes.
Those barbecue tong teeth.
Yes.
This has been a subject of interest because bird digestive systems are interesting just in general. They're very plastic, they're very diverse. It's been thought that maybe the uniqueness of bird digestive systems might have been part of why they were the only group of dinosaurs to survive the Cretaceous extinction, that they were more adaptable to being able to adjust to new foods, things like that. But trying to understand past diets, as we've always discussed, is difficult because we don't always get gut content. We don't always get good info, like clear info as to what they were eating. This bird's been looked at to try to figure that out, and initially was thought to be a fish eater. It has a similar kind of shape to a kingfisher.
Okay.
Long bill, similar kind of skull, but follow-up on that has shown that that's just kind of superficial. They look similar, but they're not actually that similar. Comparing it to other ancient birds, some of which preserve fish in their stomachs, they found a pattern among those fish-eating birds that this bird didn't match. So it doesn't seem like it falls within fish eaters. Statistical analysis that they mentioned in the abstract of the length of the beak, body size, and tooth morphology predicts insectivory.
Okay.
An insect eater. This study is actually about the fact of two newly studied specimens of this bird, not newly discovered, but like finally looked at in enough detail, that preserve gymnosperm seeds in the belly.
Plants.
Yes, these were seemingly frugivorous. They were eating fruit of some sort.
Interesting.
Because they still had some like fleshy material on the seed. Like there was evidence of fleshy material would have been on the seed, so these were seeds inside of a fruit.
Yeah.
Gymnosperm fruit, so not angiosperm fruit, so, cousins of conifers and stuff like that.
Mm-hmm.
Which gives direct evidence for them being herbivorous in a way, and directly contradicts the previous interpretations. So it's a kind of surprising find. They concluded that it's frugivorous instead of a seed eater based on the fact that the seeds don't seem to have been beaten up.
Like they weren't chewed on.
They
or
weren't chewed
like
on
that.
or cracked. That they were incidentally eaten, so to speak. And the absence of gastroliths for crushing up the seeds, all of which are similar to J-Holornis, another prehistoric bird that we've talked about on the podcast before, indicate frugivory. So indicate that it's a fruit eater.
Yeah, that the seed is just going to pass through.
Yes.
The digestive system.
You weren't eating
the fruit is where the nutrition
seed.
was.
You were eating the fruit, and seeds got in.
Interesting.
This brings up the question of why the big teeth then?
Yeah, what's with that mouth?
Right? I don't know that they did major research on that specifically to figure out what they were for. This is more about these new specimens. But due to the fact that they resemble hypercarnivory, like they resemble carnivores going after prey, they even compared it to other meat-eating dinosaurs, like similar carnivorous teeth. The one suggestion one of the researchers gave is maybe they are indeed weapons, but against their own kind.
Oh,
Similar
sure.
to the way hummingbirds will use serrations and hooks on their beak to fight each other off from prime real estate. So it might be a competitive weapon and a territorial weapon of some sort instead of a hunting weapon.
Very interesting. That's a fun, different... Well, that goes a bit of the way to explaining the unusual shape and size of the teeth.
Yes.
That they are unusual among what we expect for birds. And it's another one of those fun reminders that, as we have discussed before, teeth serve many different purposes.
Yes.
And I as you were describing it, I was wondering, could it have been omnivorous? That it was also hunting for food, for prey? Could it have been that it was using those teeth to, like, extract the fruit or something? But the idea of it being a competitive thing also makes a lot of sense.
Yeah, and it's something we see, like, hummingbirds will have armament on the beak.
Yeah, which they're not using for feeding.
Yeah, they're specifically using that for defensive and aggression with other hummingbirds.
Very cool stuff. Well, that feels like a very fitting news piece for a plants episode, as this one ends up being. And indeed, I also have some news about plants.
Nice.
This one is more specifically about plants. This is about the composition of an ancient rainforest from Tasmania and what it tells us about ancient polar forests.
Awesome.
This is research published in the American Journal of Biology by Miriam Sladonik, and in the blog post we will share a link to the article in Live Science by Sasha Pear. The fossil site in question here is the Macquarie Harbour Formation in western Tasmania, so this is Australia. The site preserves the remains of what was a coastal rainforest during the early Eocene. So this is between 53 and 50 million years ago, which is an exciting age for several reasons. Number one, that makes this one of the oldest known Cenozoic plant assemblages in Australia. It's also at a time where this landmass was very far south. It is estimated that the paleolatitude of this site would have been about 65 degrees south latitude.
Yeah.
Quite far down towards the South Pole, which puts this forest as part of what is known as the Austral Polar Forest biome. Forests widespread around the southern part of the southern hemisphere at this time. This age also puts it during an event called the Early Eocene Climatic Optimum, which was a time during the Eocene of especially high temperatures. This was a particularly warm time period, which would be a great time to be a rainforest in polar to subpolar parts of the world.
Absolutely.
And it's also shortly before the southern landmasses completely separate from each other.
Oh, yeah.
So Australia, Antarctica, South America still have these lingering connections. So finding a fossil plant assemblage from this time gives us insights into what the southern forest biome was like, insights into what forests were doing during this particularly warm time period, and insights into what plant diversity looked like before that southern forest biome got split up into the continents as we have them today.
So cool.
So there's a lot to learn. This is a very pivotal time period. This study examined over 400 fossils from this formation. These are all or mostly compressions of plants. So the smooshed, flattened remains of leaves and shoots and stuff like that. They specifically were looking at non-flowering plants. So not angiosperms, like you said, gymnosperms and other things outside of flowering plants. They identified 12 species, including 9 conifers. So this was a very conifer-rich forest. The conifers include relatives of modern-day kauri, bunja, and wollemi pines, which are all iconic Australian trees today. 2 of the trees are new species identified in this paper. And outside of that, they also identified 1 cycad, 1 fern, and 1 pteridosperm. All of the plants that they have identified are most closely related to modern species in Australia, New Zealand, and South America. So predecessors and ancient relatives of the modern forests and vegetation that we see in southern continents. They note a couple of really interesting things about this forest. For one, it's quite diverse. Not only are there a dozen species identified here in this paper, but the leaves come in multiple different morphologies. There are both large and small leaves, which the authors point out could indicate a complex canopy, possibly a more open canopy. Plus, there are ferns and cycads that would have lived in the understory. So this was a complex forest.
Yeah.
There was a complex structure here, which is, we might think that being so far towards the pole, where not only are you going to get potentially temperature extremes, but light extremes, that you have long periods of darkness and long periods of perpetual light, plants might struggle in that.
Absolutely.
So finding a complex forest structure is interesting because it suggests a thriving, lush environment.
Well, and also that different modes of plantitude, like being a plant, were successful there.
Yeah.
Because you find environments where it's like, just tall trees, pretty much. Like one shape of tree is what this forest is made out of. You don't always find that variety. So it's interesting that you do find it in this extreme southern area.
Yes. They also noted some potential adaptations to life at high latitudes. High meaning a big number, in this case farther south. Some of the species showed larger leaves, which can be an adaptation to survive in environments like this. Flattened shoots, which I did not see a detailed explanation of why that is an adaptation for high latitudes, but they noted that that is one of them.
Interesting.
And distinctive detachment surfaces on some of the plants that indicate deciduousness.
Yeah.
That's where you're dropping your leaves off. So these would have been plants that lost their leaves, which is a great way to survive either when it gets cold and you can't survive very well, or when it gets dark and what do you need leaves for?
Yes.
So there are a number of signs of potential adaptations to this living at a latitude where forests like this cannot survive today because it is far too cold if you get down that far south.
Very cool.
They also note that the composition of the plants in this assemblage are similar in many ways to fossils of a similar age from Argentina, which supports the idea of a continuous polar forest biome across the southern hemisphere during this early Eocene warm period. That much like we think of today, right here in North America and the northern hemisphere, there's pretty much continuous forest across, like our latitude where we live. This was a time where you would have had a similarly continuous forest biome almost at the South Pole.
Which is awesome. Like, it's both cool just to be able to find evidence for a giant forest of the past. Like, that's cool in and of itself. The fact that it's in such a unique area and during a transition or key time for understanding the history and how things came to be the way they are today with forests and the in the southern hemisphere. That's just, it's so exciting when we find one of these sites that's just like, this has got a lot of answers and a bunch of answers for questions we probably aren't even asking yet. Like, yes,
well, you find a site and you go, we're going to learn so much.
Yep.
This site is going to teach us so much.
It's going to be so good. That's fantastic.
Yeah.
My next news is not plant related. It is Australia related, though. This is another Australian fossil. This is a coelacanth with some features that allowed them to look at coelacanth evolution and found some interesting patterns and trends in their evolution. This is research by Alice Clement et al. In Nature Communications, and the article is by Stephanie Papas in Live Science. So coelacanths today, we have two species. They are some of our last remaining lobe-finned fish, meaning they have fleshy parts inside their fins.
Yes, episode 83.
They are often called living fossils because they resemble very heavily very ancient, going back to the Cretaceous fossil coelacanths. Like, they look basically the same at a glance. So they've gotten that moniker of living fossil. They haven't changed, quote unquote.
Episode 90.
Yes. This is a newly identified species. This is, and I'm going to do the best we can with pronunciation, Ungamgawi werngarai. This means ancient fish, and it's in the language of the First Nations Guniandi people.
Cool.
This is a small coelacanth, only measures about 7.8 inches or 20 centimeters long.
Oh, little guy.
Which today's coelacanths, for anyone who doesn't know, are actually quite big. They're like 6.5 feet long, like 2 meters.
They're big fish.
They're pretty sizable fish. This is a smaller ancient member. It was found in the Kimberley region of northwestern Australia in the Gogo Formation, which dates to the late Devonian, 385 million years old, which at that time would have been a thriving reef, and one of the researchers noted it as being Australia's first Great Barrier Reef.
Cool.
This is one of their first great reefs. This coelacanth is also extremely exciting because it is 3D preserved.
Ooh.
It is not flattened. It is one of the best anatomical looks at coelacanth history. So it is very exciting for understanding the history of their anatomy, which is what they wanted to take a look at, is getting an idea for what has been the pattern of coelacanth evolution, partially in response to the living fossil accusation of how much have they and have they not been changing.
Right.
They did a comprehensive analysis of the group to look at the phylogeny, where it falls out, but also evolutionary rates for different aspects of the fish, and the morphological disparity, meaning how many different shapes of coelacanth were there throughout time. This one seems to fill an interesting transitional gap. It has anatomy that's a little bit like some of the earlier, more primitive species and the modern species, so it is a bit transitional in its features. They found a couple of different interesting patterns. There's a major shift in disparity in their morphological shapes between the Devonian and later after Devonian coelacanths. And that since the mid-Cretaceous, they have shown basically no change in what they described as discrete characters, things being like overall large-scale features, the body shape and things like that, have been effectively unchanged since the mid-Cretaceous. But during that time, other traits, ones they called meristic or countable traits and continuous traits, were continuing to evolve, and they made particular mention of the jaw and skull continued to evolve that time. So parts of the animal were indeed changing, and certain traits were indeed changing, but some of the zoomed out view features do seem to be extremely stable during that time.
Yeah.
The way they put it, which I thought was really good, is if researchers had only had skulls of fossil coelacanths to go on, they would have never called it a living fossil.
Yeah.
That the skull is so different, there would be no way you'd call it a living fossil. But because we had the whole body and that was what we noticed first, it caught the attention first. But the thing making all the headlines on this research is the fact that they looked at what seems to be matching up with the rates of evolution in coelacanth history. And they found that out of all of the environmental factors, tectonic activity was the most comparable to matching up with their patterns. More so than ocean levels or water temperatures. When the continents and tectonic plates were moving faster and more aggressively during more active tectonic periods, coelacanth evolutionary rates were more exaggerated. And they propose that could be that you're creating new environments, but also splitting up populations more frequently.
Yes, that's making a lot of shuffling around of where different populations live, which provides opportunities for diversifying.
Yes, and diversify or die. If your environment shifts or is going away, you need to respond or you are going to go extinct.
Yes. And even if it doesn't move where you are, it could open the door for new foods to come in or new predators to come in.
Or new currents, that will change the water conditions. So yeah, just that shifting seems to be syncing up with their evolutionary rates, which they point out, just could mean that tectonics could be a good thing to look at more frequently for evolutionary histories and groups as to whether or not it's fitting the patterns you see.
Yeah, that the tectonically active times might be where we're seeing extra diversification of certain groups.
Exactly.
Very cool. Makes sense.
Yeah.
That's a very cool interpretation.
Yeah, I like this. I liked the skull quote a lot.
That's very cool.
That made me very happy.
That's really neat.
I like this news.
Well, I've got one more bit of news, and remember how my last news was about a cool new fossil site that teaches us a lot of stuff about a time period that we don't have a lot of insight into?
I do.
This news is about a cool new fossil site that teaches us a lot of stuff about a time period that we don't have a lot of insight into. But totally different part of the world, totally different time period, and some cool different fossils to look at. This is a fossil site from the early Pennsylvanian, so almost as old as the fish you were just talking about, and it's from here in the United States. This is research by Richard Knecht et al. In Nature Communications, and we will link in the blog post to an article in the Harvard Gazette by Anne J. Manning. The Pennsylvanian period is, here in North America, one half of the Carboniferous period, a very important time period for the evolution of life on land. Lots of important stages in the evolution of vertebrates on land and plants and insects and all sorts of stuff. We have tons of fossils from the Pennsylvanian that come from coal beds. Again, very popular here in North America. Most of the information tends to come from the middle part of the Pennsylvanian, with less from the early part of this period. So there's this part of this important time frame that we have significantly less evidence. This study describes a fossil site from the early Pennsylvanian, around 320 to 318 million years ago, older than most of the other Pennsylvanian sites. This is called Lantern North in the Wamsuta Formation of Massachusetts, in the northeast of here in the United States. This deposit preserves a seasonally wet forest environment, which is also different from most of those coal beds, which are swampy, ancient environments. So it's got a bunch going for it that distinguishes it from other well-known Carboniferous sites. It is also a lagerstatte, a site of exceptional fossil preservation, including both body fossils, but also and mostly trace fossils. Footprints and traces left behind by animals in the environment. These include a bunch of different stuff. There are footprints of temnospondyls, so early amphibians, synapsids, the ancestral relatives of mammals, and early reptiles. Not only walking traces, but also swimming traces. And there are lots of traces of invertebrates, including arachnids, so things related to spiders, myriapods, which are your centipedes and millipedes, and insects, both wingless and winged insects, and both adult and nymphal insects.
Wow.
So tons of diversity in the trace fossils. There are also, they report in the paper, some places where you can see mud cracks in the sediment and raindrop imprints, which, super cool. But also that tells us a bit about this sediment was exposed to the air from time to time.
Yes.
That this wasn't a permanently flooded surface. In addition to all of the cool fossil animals, there are also tons of plants from this ancient forest. They mentioned that they are now from this site are known over 131 plant species, including 83 different leaf morphologies, different shapes of leaves. So tons of diversity of plants. These include ferns, lepidodendrons, which are your scale trees, sphenopsids, which are something else, and others. And the leaves also show direct evidence of the interaction between different organisms in the form of damage on them.
Yeah.
There are galls, which are left often when insects get inside a branch. There's damage on the leaves from piercing and sucking mouthparts as insects were pulling fluids and stuff out of the leaves. And there's also damage on the leaves that they interpreted as evidence of oviposition.
Cool.
Of insects slicing into the leaf tissue with their ovipositor to lay eggs inside the leaf. They noted that these are, again, not only information into the ecosystem and the ecology of the things that lived here, but some of the earliest known evidence of these behaviors. They mentioned in the abstract that this might be the oldest direct evidence of insects depositing eggs inside leaves, of ovipositor activity.
That's so awesome.
It's super cool. And one more thing to mention, I do want to note, I had already picked this news and taken most of my notes when I noticed that they also mentioned that some of the leaves are associated with fungal rhizomorphs.
Hey-o.
That is those, we talked about this in the last episode, those cords of mycelium that fungi sometimes develop. They noted at least 3, I think, different examples of leaves with these fungal cords growing on them.
Cool.
Very cool stuff. This is mostly this report is look at all this cool stuff in this fossil site. Very much like the Tasmanian site that I mentioned earlier. This is a report on a site that has a lot of potential to teach us about the early evolution of the plants, various invertebrate groups, various vertebrate groups from this time period where we typically don't get a lot of representation of terrestrial ecosystems.
Awesome. It's very exciting because a paper like this often feels like the brochure for this new fossil site, where it's like we're not doing like a bunch of conclusive analyses of like what does this tell us about the balance of this ecosystem?
Right. We're not making a detailed description of this new species or anything like that.
We're not trying to figure out what does this tell us about the overall evolution of one of the groups here. We're just documenting and cataloging and putting together the overview and list and census of this fossil site.
Yes.
And it's fun because then you could just list all the cool stuff that's there, but also then it is a way to say, hey, researchers,
this exists.
Now you can be aware. So if you go, hey, I was studying spider tracks and
yes.
I didn't know this site existed because this hadn't been published yet. Now it has, so I can add it to my research. Like,
yep.
I always get excited when we get a big collection of, here we listed out a whole bunch of things we found at this fossil site, because it's like a bunch of researchers just went, ooh,
yes.
You found that there? Oh, that's a different area of the world than I had for any of the specimens I was looking at. That's so cool.
Also, this paper and the last one, the Tasmania site that I talked about, are both open access.
Yay.
So if you follow the links in the blog post, you can make your way to the paper and see all the pictures of these plant and animal fossils.
That's the other thing that's fun about these papers, is it's usually just full of pictures.
Yep.
And they're so much fun to look at.
Yes.
Awesome.
That's the news, and this was a very plant-filled news set, which is very fitting. Which means it's time to return to the topic of fungi. Last episode, we did our big overview of fungi and their evolutionary history. This episode, we dive into arguably the most famous and important relationship that exists between fungi and anything else in the world, fungi and plants. After this musical interlude, we will get into that, joined by our favorite paleobotanist, Dr. Ali Baumgartner. Stay tuned. Hello, Ali.
Hello, David.
Welcome back to the podcast. Thank you so much for being here for this, the second of our fungus extravaganza trilogy to celebrate our 200 episodes.
Episode 200, part 2.
Oh, yes. It's 200 part 2.
Episode
Yes,
200
exactly.
part 2. Episode 201.
Yes, exactly.
Ali, how are you?
I'm so excited to be here. I am trying to survive the beginning of the semester.
Ooh.
Best of luck.
Yeah, we're to the point where all those meetings we will have in the future is now.
Yeah. Tomorrow Allie's problems are today Allie's problems.
Correct.
So when we were planning this fungus stuff, way back when we knew that we would end up having an episode about fungi and plants, partially because it's a big topic, partially because, as our listeners will already know at this point, it was requested specifically many times.
Yep.
And so we almost immediately reached out to you and said, hey, do you want to talk about fungi and plants? And thank goodness you said yes, because it's a lot of work to do 3 episodes. And this way, we only had to do 2.
This is true. And I do like any excuse to talk about plants, even if it's only kind of adjacent.
Yes. Even if it's a co-star.
Correct. Still counts.
Yeah, the plants get second billing
Yes.
this episode. So last episode, episode 200, part 1, episode 200, we did a big overview of how fungi work, what their fossil record generally looks like, but we didn't really go into any detail about the relationships between fungi and plants, which are a very big deal. In the last episode, we talked about how most fungi get their nutrition from plants. Typically, they are saprotrophic. They are decomposing plants. They are feeding on decaying plant material. This episode, our plan is to go a step beyond that and talk about the interrelationships between living plants and fungi. Ali, there are several major categories to focus on here. We'll talk about symbiosis and parasitism and other stuff. I'd like to start with symbiosis. So when you're ready, take it away.
All right. Symbiosis. Let's go. So the fun thing about plants and fungi is that they have been historically stored together in herbaria. The herbarium I work with, I think we have maybe the second largest collection of fungi in the country.
Cool.
Oh, wow.
We're kind of a big deal.
Yeah.
So they have been studied together for a long time, and that makes sense because they're literally attached a lot of the time.
Yes.
So if I were to ask most people, just shot in the dark, give me a name of a symbiosis between plants and fungi, I bet you most people could come up with mycorrhizae.
Mm-hmm.
Because that is something that got covered in a lot of, like intro bio, high school biology. So somewhere deep in many people's mind is the word mycorrhizae. So let's talk about what that actually is. So it is typically a mutualistic relationship, right? So both partners are benefiting, typically. Sometimes, the scales can shift. It's most common in vascular plants, but it's also found in non-vascular plants, this relationship between plants and fungi, which is kind of surprising given that it's normally associated with roots. It's right in the name.
Yeah, exactly.
Yeah, mycorrhizae means fungus roots.
Yes.
This
Exactly.
is a tight connection between fungi and the roots.
Absolutely, yes. So it plays this huge role in plant nutrition and in soil biology and soil chemistry, because there are some people who argue you can't have soils until you have roots, and the relationship between roots and fungi is very close. So in many ways, you can't have soil until you have fungi there to help you make the soil.
Yeah, mycorrhizae usually consists of the roots of the plants with that fungal mycelium growing in and around those roots.
And it's really important. Like, plants really function at the level that they function at because of the fungi. So the plant photosynthesizes and it shares the products of photosynthesis with the fungi, right? It's giving it carbohydrates, these sugars, so that the fungus can survive. And then the fungus supplies the plant with water and nutrients, most specifically phosphorus. That is the most important thing that the fungi is getting for the plant. So the rate of phosphorus flowing into mycorrhizae can be up to 6 times the rate of plant roots alone.
Wow.
Whoa.
And the reason for that is because of fungal hyphae.
Yes. Yeah. Last episode, we talked about fungi grow in this sort of net of filaments called hyphae, and that gives them an extraordinarily big surface area.
Yes, and they are very fine. So you can fit hyphae in places where roots can't fit.
Yeah.
Oh, interesting.
So you can kind of get into more places and suck up all those nutrients. So there are a couple different types of mycorrhizae that I will be breaking down, but in general, plants will give 15 to 30% of the products of photosynthesis to their fungal partner. And in return, the fungal partner will provide from 80 to 86% of the plant's phosphorus and nitrogen.
Wow. All right.
Wow.
In addition to nutritional benefits, the fungus could also provide non-nutritional benefits, including secondary metabolites, which I'm going to talk about more in a little bit. So secondary metabolites I've talked about previously when we were talking about plant defenses. They are things that the plant makes that don't relate to reproduction or growth.
Right. All the other stuff.
Literally everything else. So fungal secondary metabolites can protect against pathogenic fungi, nematodes, bacteria, any little microbes that you don't want getting into the plant. So there are 2 main types. There are many subtypes. I got too deep in the weeds and realized that I needed to get out.
Too deep in the roots.
Too deep in the roots. So I'm only going to talk about the 2 most common types of mycorrhizae.
Put in your requests now for a mycorrhizae episode.
Oh,
absolutely.
And we'll
do way more detail.
You could go ham on this. So the most common type of mycorrhizae, absolutely, there is no contest, is arbuscular mycorrhizae. So the arbuscular mycorrhizal fungi. So AMF, it's often abbreviated as. It is so common. It is found in roughly 80% of vascular plant families, including a lot of agriculturally important groups. And I kept seeing a quote floating around that it is quicker to list the plants that do not form the endomycorrhizae than those that do.
Wow, yeah.
This is one we made brief mention of this
Yes.
in the last episode, that fungi and plants obviously have lots of close relationships, just like plants and animals, just like fungi and animals do. Mycorrhizae specifically, like if you've met a plant, you have almost certainly met a plant with mycorrhizal fungi.
Absolutely. It's fascinating. One of the groups that many members of or one family where many members of the family do not have arbuscular mycorrhizae are the brassicas,
Oh, interesting.
right? So like cauliflower and broccoli and all the cabbage, all those friends apparently don't have arbuscular mycorrhizae, and it was notable, this fact.
Huh.
Yeah, bunch of weirdos.
They are.
It's got the same thing of like what we talked about, like animals have gut microbes and stuff. Like some just live in you, but others are doing important jobs. When we lose that, it's bad.
Yeah, this is like hearing that there's a group of animals that don't have gut microbes.
Exactly. Like that's very weird.
Don't worry. They have other microbes. We'll get to that later.
Yeah, we sure will.
So the most important thing about this arbuscular mycorrhizae, two things. One, it penetrates the host cell walls. So it is actually integrated into the cellular structure of the plant. And it forms these structures called arbuscles, which are basically these like tree-like branching structures that are just another way to integrate into the plant. They often also have vesicles, these like globular structures. And historically, this was called vesicular arbuscular mycorrhizal fungi, VAM, which is that's what I learned when I was in school. But not everyone has vesicles, but everybody has arbuscles. So
Sure. We're being inclusive.
We're being inclusive here.
So these are, you've got your fungal hyphae, the threads that are all over these roots. And then from those hyphae, they grow into the cells.
Which is a level of symbiosis that's insane.
Yes,
Yes.
Inside the cell symbiosis.
That is not the usual case.
And then they form those arbuscles, that sort of branching structure inside the plant cell.
Yes.
And if you're going to get inside any cell, a plant cell is a very impressive cell to get into.
Yeah.
Famously difficult to get inside.
Yes.
I also can't help but keep picturing, if anyone remembers those like creepypasta Garfield comics and things that took over of there being a fungal Garfield making a John Arbuscle.
Sure.
Yes.
Sure.
There you go, fan artists.
I love that. So the fungi that form this arbuscular mycorrhizal fungi, they are from the subphylum, the Glomeromycotina, and they are obligate symbionts. They are not free living. They have only been found in these mutualistic relationships with plants.
Mm-hmm.
It is fascinating to me that apparently, despite the fact that they are obligate symbionts, right? They are only found in plant roots. They still have blue light sensors for their circadian rhythm.
Interesting.
So if they did get exposed to light, they would know what time of day it was.
Yeah. It's that lingering bit of evidence of when they used to be on their own.
Yes.
That's very cool.
Wild. So this type of fungi, they are in their greatest abundance in the tropics and in temperate grasslands, because these are found in basically any flavor, any habit of plant, right? They're common with grasses, you'll find them in trees. They do not discriminate. They are less common in very arid and in nutrient-rich soils.
Oh, okay.
That makes sense. If there's a lot of if the plants don't need the help.
Yeah.
Correct. Exactly. So that is the most common type of mycorrhizae. The less common, but still very common type of mycorrhizae are the ectomycorrhizal fungi. So these are the outside of the root fungi.
Yes.
And these do not penetrate into the host cell walls. But oh boy, do they get all up in there.
They're just cuddling.
Yes. So they'll basically get between the cells, but they don't go into them.
I love that they're in between the cells, but not getting into the cells. That has very much a I'm not touching your nucleus
Yes.
vibes.
Yes, exactly. So that makes what's called a Hartig net. And basically it is just a sheath for the root. So it completely covers it, which actually forms a physical barrier between the root, the plant, and other things that might try to get in.
It's a venom suit.
It's like
Yes.
well, I was going to say it's like a koozie, but
No,
that is better.
It's a venom suit.
It is a venom
I
suit.
have had the venom analogy in my head the entire time that I've been working on this.
Absolutely. Oh, this is the best episode ever.
I agree.
Since the Venom episode.
So
Episode 97.
it makes this dense hyphal sheath called a mantle around the root surface. And then it extends the hyphae out from that. So they can extend several centimeters into the soil, and this even occurs in the deep roots. So at least 2 meters to more than 4 meters deep.
Wow.
Right? They do not mess around. And it's funny because a lot of places I was reading are like, we associate this with shallow roots because those are the roots we can sample.
Yeah, exactly.
Yes.
That's where we looked for it and we found it and then realized, it goes 4 meters deep.
I always love reminding people that we're not good at studying things underground.
Correct.
Humans are so bad at underground. And so there's so much going on down there that we're just like, this, we found this in like this one tree that we were able to do this with. We're not usually able to find all this out.
Right. When they fall over, like,
Yes,
that's what's going on down there. Exactly.
Thanks for bringing it up.
How considerate of you. So I mentioned that the sheath acts as a physical barrier. The fungi themselves, this ectomycorrhizal fungi, allows plants to tolerate soils with high concentrations of heavy metals, high concentrations of salts, high concentrations of radionucleotides, and organic pollutants.
Well, it's just like a hazmat suit as well.
Yes.
Mm-hmm.
Absolutely.
That's awesome.
It's really impressive. I was going through and finding all of these citations. It's like, yep, they could do this. Yep, they could do this. There's a lot of studies about phytoremediation with this sort of stuff that I'm not going to get into. So typically, these ectomycorrhizal fungi are either from the Basidiomycota or the Ascomycota, right? So like the two big groups that you talked about.
Yep, the club fungi and the cup fungi.
Yeah.
Yes. And they reproduce sexually and they make fruiting bodies.
Mm-hmm.
Oh.
Which is, that's part of the reason why we associate them with shallow roots, because most of them, not all of them, send their fruiting bodies up to the surface.
Right. So you'll see little mushrooms and such growing around the base of the tree or whatever plant it is.
Exactly. So some famous examples of this are actually truffles.
Oh, really?
Yep.
Huh.
Which are in the genus Tuber, which I love. And then death caps and the destroying angel, which are in the genus Amanita, which you've probably heard of.
Fungi, mushrooms especially, get fantastic names.
They really do.
They get Final Fantasy villain
Yes.
names.
Yep.
But also just like fairy tale names. So
Yes.
my colleague, the fungi collection manager, people will send her pictures of mushrooms and like, hey, what is this? And some of them are like golden oyster mushroom, which is like, that's a fine name. But dryad saddle is so good.
Yep.
Anyway. But importantly, many of these ectomycorrhizal fungi can only form fruiting bodies and complete their life cycle if they are in a relationship with plants.
Okay.
Got it. So this isn't only essentially protecting the plants,
Right.
but the fungi can't actually complete their life cycle without the plants.
Correct. And this is really important. So I mentioned that the other type is just ridiculously common and found pretty much everywhere. This is more exclusive. So this type of mycorrhizae are found on the roots of roughly 2% of vascular plant species, especially woody plants. This is commonly associated with trees.
Okay.
All right.
So some of their best friends are the families of the birch, the beech, willows, roses, and pines, which is interesting because that's a gymnosperm and everybody else I mentioned were angiosperms.
Yep.
Yeah.
So that in and of itself is really interesting, and I'll talk about that a little bit more later. So although the plant families are quite different in the temperate and tropical forests, right? The type of plants you see in the tropics are not the same ones that you see in the temperate regions. The ectomycorrhizal fungi that you see in the tropics versus temperate regions tend to be very similar.
Interesting.
Cool. So these are groups of fungi that have themselves adapted to work with multiple groups of plants.
Yes.
Oh, and it's interesting that they haven't diversified along the same lines, even though I assume they were in those relationships when those plants would have been diversifying and stuff.
Well, interestingly, there's evidence to suggest that the greatest diversity of this type of mycorrhizae are actually in the temperate forests. Which is not where you expect to find the highest diversity, and is not where we find the highest diversity for the other kind of mycorrhizae.
Yeah, or plants in general.
Yeah.
Correct. Yeah, which is really, really exciting.
So mycorrhizae are, we did a whole episode about symbiosis. It was episode 133, and we talked about lots of symbiotic relationships that are part-time or transient, or there's a symbiosis at certain parts of a life cycle. Mycorrhizae with fungi and plants is not one of those. This is one of those. Many of these are only present in these relationships.
Correct.
Well, this feels kind of more along the lines of when we talked about parasites, where it's like there's some parasites that are so specialized to be parasites that their genome is reduced, their like biological functions are reduced.
They're only found in certain hosts.
Yep. And this is where these fungi can't do their life cycle unless they are partnered up with a plant.
Yes.
That is an extreme adjustment to how you function as an organism.
Yeah.
It is a committed relationship.
Yes.
And it means that a enormous percentage of the world's plants and fungi are inseparable from each other.
Yeah.
You only get them together. So we just mentioned the diversity of mycorrhizal fungi does not quite line up with the diversity of their host plants, which, we've talked about parasites and stuff in the past, where you can actually, in some cases, map the evolution of parasites onto the evolution of their hosts.
Oh yeah, if you give a specialist a lice, they can tell you who it came from.
Yeah, exactly. The fact that these fungi don't seem to have always done that makes me very curious about the evolutionary history of mycorrhizal relationships.
There's so much research into this for a variety of reasons, but there's so much research into this.
We are, as humans, highly invested in the healthy growth of vascular plants.
Yep.
Yes, we are invested. So one of the things that came up multiple times was specifically with the ectomycorrhizal fungi, because it has an association with pines, with a gymnosperm family, that suggests a much older association. Because pines have been around for longer. So that means that likely this was a relationship with gymnosperms before, angiosperms were even around. So the pine family dates back to like 156 million years. So we're looking at a pretty deep relationship, especially considering there's also this wide geographic distribution, right? You find this all over the place. So one of my favorite things to ask y'all is how many times you think something has arisen.
I was just thinking it.
It is one of my favorite questions. So for the ectomycorrhizal fungi, right? So that's only found in 2% of vascular plant species. How many times do you think that lifestyle evolved?
I'm going to say 2.
4.
Oh boy. Okay. So molecular and phylogenetic analysis of fungal lineages suggests that this has evolved and persisted numerous times, as few as 7 to 16 to as many as 66.
Wow. I was gonna say 20 when I thought you were just gonna ask in general, but then when you were like the smaller
Specifically
percentage,
the ectomycorrhizae.
I went, oh, okay, well then I'll do it. I should have just gone with 20.
These are the sheath making ones.
Yes.
That is really interesting because it suggests not only is this something that those particular symbiotic fungi are good at, it is something that fungi in general in those groups are good at doing.
Yeah.
Yes. Which I love. Like I said, I love figuring out or like seeing studies about how many times things have arisen. And the reason, even just understanding why this relationship exists, is kind of confusing. So it seems likely that the mycorrhizal relationship in general evolved from a relationship in which the fungi was taking nutrients from the plant in general into a symbiotic relationship where the plant and the fungi are exchanging.
Okay.
But like the nature of that initial relationship is still kind of up in the air. The specifics, like in general, it used to be one-sided and now it's two. So whether or not it evolved from a parasitic interaction that developed into a mutualism, or whether it evolved from a saprotrophic fungi that became endosymbiotic.
Right. That they were taking nutrients from decaying plant material,
Yes.
and then that transferred over to living
Mhm.
plants. Or they were stealing nutrients from living plants detrimentally, and then that developed into a two-sided relationship.
Right.
And with that many different evolutions of this feature, it wouldn't surprise me if multiple different origins
Yep.
Right.
are all true.
Right, exactly. It's biology. The answer is always complicated.
Mm-hmm.
So, in general, by the Middle Devonian, all of the major groups of land plants had roots. So, by that point, it seems pretty certain that mycorrhizae were probably pretty well established in whoever had roots at the time.
All right, this is about 400 million years ago.
Mm-hmm.
Correct. And in addition to some fossil evidence, which we'll talk about in a bit, the fact that we do see mycorrhizae associated with things that don't have roots today suggests that that's something that they probably could have been doing for a while. And the modern distribution of mycorrhizal fungi appears to reflect an increasing complexity and competition in root morphology, which is associated with the dominance of angiosperms in the Cenozoic. Everything
Gotcha.
is related to the dominance of angiosperms in the Cenozoic.
Yes.
Yep.
Episode 57.
And there are ecological implications for the absolute dominance of the arbuscular mycorrhizae. Plants of different species can be linked into this mycelial network, right? You can just have all of these plants that are basically talking to each other, right? You're sharing nutrients. And it is fascinating. You can have one plant that is providing the photosynthetic carbon to another plant via this mycelial network, which just feels futuristic in a very old-fashioned sort of way.
That's definitely the most sci-fi
Yes.
aspect of mycorrhizae, in my opinion. Like it's something from the Avatar. Like that's
Yes.
The Pandora plants have a neural net that they communicate with each other.
Also, the other avatar in the swamp.
Yep.
So you've got these mycelial networks surrounding the roots of a plant that can grow out and connect, be part of the mycelial network that then extends into another set of plant roots. But they can also do that cross species.
Yes.
So the same mycelial network can be attached to multiple different species of plants, which not only is really interesting, again, because that these symbionts are not specific to a particular host species, but it also means that, these different species of plants are now also existing in a symbiotic relationship with each other.
Yes.
Yeah. It's, as we often make the point, that's a ecosystem, and like players in an ecosystem functioning and interacting with a way that is wholly alien to the way animals
Absolutely
do it.
Yes.
So that it's really hard for us to wrap our brain around the actual complexity of what's going on.
So thanks to these fungi that are underground, if you cut down a tree in the forest, all of the other trees nearby know that happened.
Yes.
Like a tree over there, that pine tree knows that a tree has died.
And the fascinating thing about this whole conversation, too, is that oftentimes when information like this gets shared in, like, popular science, there's that temptation to go just a little bit further and kind of like speculate about like intent in that sort of thing. And I just want to point out that fungi are weird enough without trying to anthropomorphize them.
Right? I was going to make a joke of like, this makes you want to ask who's really in charge, the forest or the fungi. Because, it's really hard not to think about like, what are you doing? So, well, that's not how it works.
Well, and the reality is that they are inseparable.
Yes.
You cannot distinguish between where obviously their bodies are different, but the process that is going on, the biological processes, there is not a hard line between what the plant is doing and what the fungus is doing. It means that a forest, especially in like the temperate regions, the tropical regions, a forest is composed of a network of trees and branches above the surface and a network of roots and mycelia below the surface.
Yeah.
Yes. Exactly.
Cool. It's like an underground version of the canopy.
Yes.
There's a canopy on both sides.
Except for you have crown shyness in the canopy, so they're not touching. But down in the below the ground, they absolutely are holding hands.
Well,
That's
and
true.
that was precisely what sticks in my brain about this relationship and about this dynamic, and why I was saying like it's hard to wrap an animal brain around this, because we talk about the fact that plants compete, that their branches shy away from each other and they try to overcrowd one another and get sunlight. But then underground, they seem to all be holding hands in a way. So those two truths seem to disagree with each other, but they're obviously both happening in the same forest at the same time, which just makes it's very hard to comprehend from an animal point of view what's actually happening. It's the SpongeBob thing where we lift the shirt and it's the heart, but it's mycorrhizae.
Yes.
We're like brothers, only closer.
So, are there mycorrhizae fossils?
Yes. I will go into more detail.
Moving on. Please, that was a rhetorical question. I did a whole bunch of research for the last episode. I know there's mycorrhizal fungi.
I'm glad you asked.
Yeah, absolutely.
I had no clue.
Allie, please tell us all about the mycorrhizal fossils.
Tell me.
All right, this is for you, Will. So as David mentioned in the last episode, fungi do fossilize, just they're not the most common fossils. It's the same way that people like, oh, there are no plant fossils. No, there are plenty. We're just looking at the wrong ones. And as David mentioned, the paleobotanists also have all of the fungi fossils.
That was a quote from, I forget what reference it was, but it was one of my references that said, one of the issues is that the mycologists have all the knowledge about fungi, but the paleobotanists have all the fossil fungi.
So accurate. As soon as I heard that, I was like, checks out. Because palynologists are also the people who study fossil pollen are also the ones who have all of the like fungal spores. So it's a whole thing. Anyway, and also as you mentioned in the last episode, the Rhynie chert is just riddled with fungi.
Yes.
And it makes sense, right? What is going to be good preservationally for plants is going to be good preservationally for fungi. We have similar needs. I say we. I'm still not a plant.
Yeah, sure. As much as you may wish.
But you are part fungi.
I am. This is true.
So the Rhynie chert, for those who didn't listen or forgot, is a fossil site in Scotland from the Devonian that is extremely famous for plant fossils. And also, I made a point to plant the seed, as it were, of talking about the Rhynie chert in the last episode, because I knew it was going to come up in this episode.
Oh, absolutely. As soon as I heard it, it's like, yes, thank you, David. I appreciate all your work.
Allie and I are in a symbiotic relationship working on this episodes.
It's true.
The mycelium net hasn't made it to me yet. I don't know what's going on.
So the discovery of Aglaophyton major, like that's the one that people talk about with like, this is the earliest thing that, the earliest fossil showing this arbuscular mycorrhizal fungi and early Devonian land plant from the Rhynie chert because of course. And this is really exciting because this puts this relationship more than 400 million years ago.
Which is a lot.
Basically, once plants get on land, they've basically have this relationship with fungi. Like this is from the beginning. They've been besties forever.
Well, it's the buddy system.
Yeah.
If you're going somewhere new,
It's dangerous
Take
to
a
go
buddy.
alone.
take this.
Yeah.
Just bundle of mycelium.
So the earliest plant, it sounds like essentially the earliest fossil plant roots have fossil mycorrhizae in them.
Yep, pretty much.
So this relationship is there as far as we can tell the whole time.
Yeah. As far as we can tell, this goes back to the beginning.
So cool.
Yeah. And the cool thing about this too is that it is in some ways easier to see the arbuscular mycorrhizae than the ectomycorrhizae, because the arbuscular mycorrhizae is fully integrated into the cells of the root.
Yeah.
Yeah.
You have this internal structure that you can look at and point to, like, yes, those are the things that we are looking for. That means that this is here. So yeah, basically from the early Devonian, once plants are like getting themselves established on land, we have these interactions with fungi, which is really exciting.
And in the Rhynie chert, you get, in many cases, such good preservation of fossils that you can see cellular details.
Yeah.
Yes.
And there are fossils of, here's a microscope, an electron microscope image of the cells of this fossil plant, and inside some of those cells are these fungal arbuskels.
Yeah.
So the arbuscular mycorrhizae are way more common and unsurprisingly seem to have arisen earlier.
Okay,
That makes sense.
cool.
Some of the earliest evidence for the ectomycorrhizae is way more recent. Like 50 million years ago. More recent.
Wow.
That's the opposite. Not only is that the opposite order that I would have just assumed,
Mhm.
that you would have started as a sheath and then started making intercellular invasions, but that's also just a huge gap.
Yeah.
So that they're out in my brain, they're out of order and way too far apart. I disagree with all of this.
This is just the fossil evidence. So
Yes,
the
exactly.
molecular is saying a little bit different, and I'll talk about that when I talk about terrestrialization in general. But yeah, it's a way more recent relationship than you would expect. So there's this cool fossil. Again, it's about 50 million years old, and it is from a pine.
Nice.
And you can see this sheath, this hearting net that is on the outside of the root, as well as some of the hyphae that are coming off.
That's
very cool.
That's nice. Awesome.
Yeah. So those are some of the kind of charismatic fossils, because let's be real, these are not the most exciting looking fossils.
Sure.
They are really important for life on Earth, but in terms of something to look at,
The squiggles.
yes.
Yeah, they're microscopic squiggles.
It's
Yes.
just, doodles.
Absolutely.
This is an interesting one because, especially with the arbuscular mycorrhizae, which today, as you mentioned, all over the planet, almost all land plants have them. We have fossil evidence going over 400 million years ago, which means whether we have direct fossil evidence or not, if you find a fossil of a plant, it almost certainly had arbuscular mycorrhizae.
Yes.
It's the same way as if you look at a plant today. You don't have to go look at the roots to know if it has if you take a guess, you are almost certainly going to be right that it has those mycorrhizae.
Absolutely. And that's not the only way that you can be a fungus living in a plant.
Oh.
That's true. So mycorrhizae are the famous symbiotic relationship, vascular and even non-vascular plants with fungi. I'm sure there are other symbiotic relationships that are worth mentioning.
Oh, absolutely. I want to introduce you to the fungal endophytes, which are fungi that live in plants.
All right.
That's all that means. I love the names. So the difference between a fungal endophyte and mycorrhizae is whether or not the fungus is bringing nutrients from outside of the plant into the plant.
Okay.
right? So mycorrhizae, right? They're going on the little adventure, sending their hyphae out there, picking up nutrients and bringing them into the plant. Fungal endophytes are basically doing everything but supplying the plant with nutrients. So the plant is still giving it carbon. It's still a mutualism, but the endophyte is primarily giving chemical protection.
Okay, gotcha.
Those secondary metabolites that I said I was going to talk about, I'm going to mention them in a bit. So these also do not go into the cell walls. So these, again, are just going around all of the cells, basically wrapping them up and having these hyphae that are extending out. But they will penetrate the plant's embryo and grow along the seeds to infect the new plants that will grow from the seeds. These are vertically transmitted.
Oh, wow.
Yeah, so these are fungi that live in the plant tissues. And then when the plant reproduces, the fungi are in those tissues so that it hitches a ride with the seed.
Yeah. Trojan horse is the first thing that comes to mind, but it was
inside to begin with.
But it's like, but instead of when you jump out, it's a surprise party. It's good.
Yes.
You want these in there.
I've been here the whole time. So this is mostly a mutualism. So the endophytes help the health and the survival of the host plant with issues such as pathogens and disease, water stress, heat stress, nutrient availability, poor soil quality, salinity, herbivory. Like, they are there to help. So they receive the carbon from the host plant, and then they can do all these things. However, if the plant is stressed, then they are in prime position to become a pathogen.
Sure. I was just about to say, this is very similar to, if we think about like our gut
yes.
microbes.
This is basically exactly what this is.
Yeah. These are microorganisms living inside our bodies, assisting with certain processes that go on. But if you start getting stressed or sick, some of those are opportunistic and they will develop an infection.
Absolutely.
That you've kind of got this ticking time bomb waiting inside just most plants that is great most of the time unless it's not.
And plants do sometimes have ways of like, oh, we're stressed. Okay, eject the endophytes.
Yeah.
But it is really cool because the presence of endophytes, they can so extensively colonize a plant to competitively exclude potential pathogens. There's no room.
Yeah.
So they protect against diseases in the plant in part by just taking up so much space that other diseases can't get in.
Yes.
It's already infected with this fungus.
Occupied. This plant is claimed.
Yes. Exactly.
More mycelium than tree now.
I mean, kind of, yeah. So the presence of these endophytes basically changes the chemical composition of the plant. And so this is a deterrent to insect herbivory, ungulate herbivory, oviposition by adult insects. It's helping to exclude things you don't want there. So a famous example of an endophyte is ergot.
Mm-hmm.
Are you familiar with ergot?
A little.
Nope.
Oh, well, this is a gift for you. So it is a fungus that infects rye, and if you consume it, it can make you go mad. It is thought to be the reason for the Salem witch trials.
Oh, okay.
Uh-huh. So it's very funny. We were making a exhibit at the Natural History Museum here, and we had a moment of like, oh, we should put ergot on display, and then we realized that's a bad idea. We should not put ergot on display. Our ergot is in our controlled substance cabinet that we can't access according to the DEA. So we did not.
Do not eat.
No, do not eat. There is at least one, if not more, species of endophytic, fungi in nearly every individual land plant.
Okay. Wow.
Yep.
And often You can have multiple specialist fungi infecting one individual plant.
Huh.
So it's not just that the roots are covered and infiltrated with
Yes.
fungal mycelium. It's just the whole plant.
Yes.
Yeah.
Plants are mostly fungi.
Yeah.
Yeah.
But this kind of has that vibe. I can't remember what it is. Is it tuberculosis? There's some lung bacteria or infection that most people have. It's just bound up in scar tissue. I can't remember which one it is, but it's like there's things like that where it's like, yeah, most people are infected with this. It's just the version our body can handle. So yeah, you have it in you.
Sure.
It's just not doing anything negative right now. So
Right now.
yeah, like if something goes wrong in the situation, it can
Mhm.
become a problem. But most of there are diseases out there that most of humanity are currently infected with. It's just not being a disease right now. And this kind of has that feeling where it's like, yeah, they're all infected, but mostly it's in the good way.
Yeah. So these fungal endophytes are one of the places that we are looking for new drugs. Because think about it, right? Antibiotics, that comes from penicillium, comes from a mold. So endophytes, like I mentioned, produce these secondary metabolites, these compounds that have nothing to do with reproduction or growth. They can include, I'm just going to give you a big old list, I'm so sorry, antibacterials, antifungals, antivirals, antiparasitics, cytotoxic, which means it's toxic to cells, neuroprotectives, antioxidants, insulin mimetics, meaning that it copies or mimics insulin, anti-tumor, and immunosuppressant properties. So
Very
basically, anything you want in your medicine, you got to look at a fungus. And
Yes.
these compounds can also be useful against pathogens that are antibiotic resistant. Because you know where the antibiotics came from? They came from fungi.
Yeah.
And one of the cool things about this is that many of these secondary metabolites we thought were produced by the plant.
I was wondering.
Yep.
But they were produced by these endophytes inside the cell or adjacent to the cells, around the cells of these plants the whole time.
Yeah.
Very cool.
Mhm.
Do we have fossil endophytes?
We do.
I also knew the answer to that question.
Yeah, I didn't.
So I read a whole paper that was talking about like the preservation of endophytes in fungi. And basically, the thing I found most interesting, I'll go into other bits, but the thing I found most interesting is that the fossil record is riddled with leaves.
Sure.
And we know that endophytes live in leaves. But we don't really have any evidence at all of fossil endophytes in leaves.
Huh.
Interesting.
And the primary reason for that is going to be so obvious to you. It's because they're compression fossils.
Yep, that was my guess. Yeah, you don't have the three-dimensional
Right.
preservation
You don't
of the
have
tissue.
the inside of the leaf where
Yes.
the stuff is.
Yep.
But also, you have the bias, the collection bias, right? We want the nice leaves. We're
Yep.
not taking the gross like ones, which are more likely to have these endophytes. And so there's this temptation to think that, oh, this is a more recent development
Mhm.
because we're not seeing it in this very common thing in the fossil record. And no, you got to keep in mind these other aspects that are going to be contributing to this. The other thing, too, that you have to keep in mind with kind of all fungal fossils, less so with mycorrhizae, but with most fungal fossils, is figuring out the timing. Right? Is
Yep.
this something that was living inside of the inside of the plant? Or is it breaking it down because they're decomposers and that is what they do?
Yep.
Yes.
It's the scavenging debate.
Mhm.
Exactly.
Is this bite before or after you were dead? Is
Yes.
this fungus before or after you were rotting?
Yeah, so it can be difficult to tell. And is the decay that you see just because it was decaying as part of the preservational process, or is this fungi part of it? Yes. And as plants are dying, as we mentioned, they tend to, like, these endophytes are like, now's my time.
Yes, is that like you could have one that is the same fungus
Yes.
in both scenarios.
Yep.
So yeah, this is when it was living healthily with the plant. Now this is actually it after the plant died or is about to die and is messing it up.
Yes, exactly. But There are actually fossils. So one of the oldest is from the late Pennsylvanian, so from the Carboniferous of France. Again, it's a chert because cherts are great for this. We've established because again, you can see this intercellular structure. So from this Grand Croix site in central France, there's a preserved fern pinule. So basically part of a fern leaf that has this evidence of this intracellular endophytic fungus. So we don't know who it is, but we know that there's somebody there.
Very cool. So we get almost as old as the Rhynie chert fossils.
Yes. To go back to my favorite endophyte, ergot. So it basically will form these growths on now it lives on rye, but it previously it would live on grasses. So there's evidence of ergot from the Cretaceous.
Cool.
Yes, so in amber, because unsurprisingly, that's something that you would find in amber. So you can find, so speaking of Cretaceous, you actually can find evidence of these endophytes inside leaves, but you basically have to be able to do a cuticle analysis. So unless you have the cuticle, right, so that waxy layer on top of the leaf preserved, then you might have a chance. But man, messing with cuticle is chemically terrifying to me. You have to use hydrofluoric acid.
Ooh.
It's terrifying. Want nothing to do with HF. So we have evidence of this from, say, the Carboniferous, but it's labor intensive, and the paleobotanists are probably distracted by the plants.
Yes, exactly.
So we talked about, we're sort of working our way in a direction here.
Yes.
Where we've been talking about symbiosis, but we have already started touching upon the fact that some symbionts kind of can become an infection or become detrimental.
Yeah, they're fair weather friends.
Exactly. And I also know that another majorly important category of interactions between plants and fungi are straight up parasites and pathogens.
Yes.
Please tell us some stuff about parasites and pathogens.
This is gonna get gross.
It's gross is a relative term. So when we're talking about plant fungi parasitism, you're probably thinking that the fungus is the parasite. Nah. We're going to talk about what the plant is the parasite. Mycoheterotrophy. So plants, as we know, are photoautotrophs. Right? They are eating sunlight in order to make sugars, and they can make their own food. They don't need anybody but the sun. Mycoheterotrophs are using fungi to be heterotrophs, to basically stealing their food. You made that for me. Thank you so much.
So this is the opposite of you can have and obviously there are tons of parasitic fungi, just all over the place for plants of all kinds. The plant is using the sun to make nutrition, as plants do, and the fungus is stealing some of that as a parasite. This is a scenario of a plant flipping the tables and saying, I'm not doing any work.
Yes.
The fungus is making nutrition from doing its normal saprotrophy or whatever, and the plant is stealing those nutrients.
Yes. So these are called mycorrhizal cheaters.
Oh my goodness.
Isn't it great? I saw that term used so many times. It made me so happy.
Forest wreckers.
They tend to be non-photosynthetic, so they don't have chlorophyll, and they get their nutrients from fungi. So they can be obligate or facultative, right? So they can be, this is the only way that I can get my energy, or they might do both or either. So like, for example, some species of orchids can shift during different parts of their life cycle. So sometimes they will be mycoheterotrophs, and they can be photoautotrophs depending. And it's really strange because the photosynthetic and the non-photosynthetic orchid species are like nested within each other. So it's not like there is a group of non-photosynthetic orchids.
Yeah, they've gone back and forth a bunch of times.
Yeah, spoilers. It's happened multiple times, guys. I am going to make you guess. Just throwing it out there.
Sure, fantastic.
So probably the most famous mycoheterotroph is Monotropa uniflora, the ghost pipe. I actually got to see this a couple weeks ago when the herbarium went on a retreat to the George Reserve, and we were wandering around the woods, and they're just everywhere. So they're a handful of centimeters, a couple inches tall. They're not very big. You'll find them in these clumps, and they look like pipes, and they are completely white because they don't need to photosynthesize. So they parasitize fungi in the family Russulaceae. So Russula are these stereotypical red mushrooms. So not the red one with spots, but just the red mushrooms. You see them everywhere in temperate forests. They are virtually impossible to recognize as species unless you're doing DNA, but they're so common. So the funny thing about this, about mycoheterotrophy, is that they are often actually epiparasites because they are parasitizing fungi that are getting their energy from plants.
Yeah, they're parasitizing the mycorrhizal fungi.
Uh-huh.
So they're kind of parasitizing two organisms.
Yes.
Yeah.
So they are getting energy from other plants via a fungus.
Yeah.
That's so cool.
They're taking the lunch money that the other plant gave them.
Yes.
Yeah.
Yes, exactly. So this is actually mostly found in flowering plants.
Okay.
Yeah, so there are other groups that can do it, and I will talk about that a little bit. But today, unsurprisingly, given the diversity of angiosperms, like most of these are angiosperms. So every monotrope, so I mentioned Monotropa uniflora, so they're in the family Ericaceae, which is the family that has like blueberries in it. There are these non-photosynthetic orchids. There's also a non-photosynthetic liverwort, which, again, this is a non-vascular plant. So these are all obligate mycoheterotrophs. This is what they do. This is all they do. There is one, possibly one species of mycoheterotrophic gymnosperm, but this is debated. People can't quite decide what on earth is going on with this weirdo, but it might be a mycoheterotroph.
Okay.
So it's a Parasitaxis usta. So it's clearly a parasite of some kind. We don't quite understand how.
Well, because there are also plants that parasitize other plants.
Yes. Oh, yes.
So it can be difficult, I would imagine, to tell. Are you parasitizing the root? Are you parasitizing the fungus that's growing on the root?
Right, exactly. Like, are you taking the direct route or are you taking a detour through a fungus? Exactly. So, and then some groups are these facultative or partial mycoheterotrophs, so like gentians that are famous for their pretty blue flowers. So some ferns and club mosses, so ferns and friends, are mycoheterotrophs as gametophytes. So remember, plants are weirdos and they have the alternation of generations. So the gametophyte is what makes the gametes, and that is a mycoheterotroph, which is wild. So not every part of the life cycle is a mycoheterotroph. It's wild.
Yeah.
So the parasitized fungi tend to be mycorrhizae or parasitic fungi that have large mycelium networks, which makes sense. That's basic. It's basically like trying to tap into someone else's like electricity or something, or poaching someone else's Wi-Fi.
Exactly what I was just thinking.
Same sort of thing. So I told you that I was going to make you guess. So the diversity of mycoheterotrophy is widespread across land plants, right? We have vascular and non-vascular seed plants, spore plants, whatever. So how many times do you think that mycoheterotrophy has arisen in land plants?
This time I'm going to say 20.
30.
Oh.
Approximately 47.
Oh,
Nice.
I had a feeling. I'm going in the middle of what it was last time, and I'm probably still not going to be high enough.
Very cool.
So as far as we know, once in liverworts, because we only have one example.
Sure.
At least 12 times in ferns. Wow. Possibly once in gymnosperms, and at least 37 times in angiosperms.
Well, this does make some level of sense that, as we've discussed, plants and fungi are both everywhere.
Yes.
So it makes a lot of sense that, fungi, there are tons of fungi that parasitize plants, but fungi are extremely abundant.
Yeah.
And so there are lots of fungi that parasitize other fungi. It makes sense that plants would repeatedly hit upon this strategy of stealing from fungi.
Yeah,
Yeah.
absolutely.
It's just, it's harder to picture because we're so used to fungi being the thing that infiltrates and grows into stuff. So the idea of a plant flipping the table is not how we typically think of plants behaving.
Yeah, plants are normally better behaved than this.
Yeah, right?
I expect more from you. So, trying to figure out the timing of this is really difficult for a variety of reasons, but mostly because when you are doing molecular clock stuff, it's nice to have literally any fossils to pin into that. So the problem with this is that it's hard to tell from the fossil record who is being a parasite.
Yeah.
Yeah, it's not quite as easy as with animals.
Yes.
Episode 102, we talked about parasite. When it's an animal parasite, it's just inside the body.
Yeah.
You
Yes.
can go, there it is, inside the other animal's body, being a parasite.
Well, and a lot
Yes.
of times you have like, yeah, that's a blood-sucking mouth part. Like,
Absolutely.
that's the
Yes.
only thing that mouth part's going to be good for.
Yes. So with plants, so one of the big indications that somebody is a parasite is a lack of chlorophyll.
Mm-hmm.
Well, that's hard to see in
Yep.
the fossil record. And so in general, the way that we do it is, okay, we found this fossil plant. It has an affinity to a family that today only has mycoheterotroph. So there is a fossil from the Cretaceous that has an affinity to Triuridaceae as a family that is known for having these mycoheterotrophs, but that's a level of just because today the family has mycoheterotrophs doesn't mean that 90 million years ago that's what they were doing. And so that makes it difficulty. And then, figuring out the rate of evolution, right, is again difficulty. But it seems like this is a relatively recent thing, which makes sense given that it is primarily found in angiosperms.
Yes.
So who knows what the liverworts and the gymnosperms and even kind of the ferns were doing. But in general, if we're looking at the angiosperms, the earliest divergence we're seeing is from the early Cretaceous, which that's about as early as you can really reasonably have this sort of thing with angiosperms. But unsurprisingly, a lot of these things are showing up in the Eocene and Miocene, which is when all the cool things happen.
Yes. Very cool. I'm excited that we got to dive into plants parasitizing fungi, which is the opposite of what we would expect. Also, it was requested among our many requests, so that's super fun.
I got you.
To give some nod to the fungal side of this, Allie, do you have any favorite fungal plant pathogens?
Ooh.
So this is something that comes up, like I said before, we humans are very invested in the healthy development of a lot of plants. Most of our knowledge, to my understanding, of plant diseases and plant parasites comes out of agriculture, like ergot, which is in rye. These are issues that we deal with when we're trying to develop crops, when we're trying to grow plants to eat.
Yeah, things that affect our meals and our sales.
Feel free to just shout out some of your favorites.
Oh, goodness. I have to remember. What is that? Oh, I can't remember the name of it. So I love ergot, obviously. But I can't remember the name of it. It is the fungus that grows on corn that is eaten in Mexican cuisine.
Oh, right.
I can't remember what it's called, but that whatever that's called, I love it. I love it so much. I don't remember your name, but I love you. Well, it's very funny because it's difficult for me to come from the plant side and be like, who do I like who infects my favorite thing?
Yep.
That's a difficult question. Because
Wheat
generally,
lacoche.
yes.
Wheat lacoche is a fungus that grows on ears of corn. I'm not running the episode, so I can Google stuff.
Yes, thank you. Yes, that's the one. I love it. I have never tried it. I would like to. I am biased towards things that I can eat.
Oh man, if listen, any Mexican listeners out there who have experience eating wheat lacoche, or anybody who has, let us know. Make Allie jealous.
Yeah, yes, that would work. That would work really well. Unfortunately, all of my favorite fungal parasites are not on plants, they are on animals, so I will not be
Yeah.
sharing them now.
Yeah. I know that there are also fossils at some fossil sites where you will have a leaf fossil or a piece of fossil wood with dark spots or
Yes.
discoloration or stuff in it that matches what we see with fungal pathogens and parasites today. So I think leaf spot disease is
Yeah.
a thing.
Yes.
So you can see those relationships between fungi and plants in the fossil record, even in cases where we can't specifically say, what is this parasite?
Yes.
What is
Right.
this pathogen?
Somebody is there.
Cool. Well, we could, I'm sure, sit here and talk about plant fungi, parasites and pathogens and other stuff forever. But we're not going to, because we have so much more to talk about in this episode. At this point in the episode, I am sure we have listeners who have been sitting there the whole time going, you're talking about the symbiotic relationships between plants and fungi, and there is an example you have not talked about once. We're going to take a short break, and after the little musical interlude, I'm going to ask Allie to talk to us about lichens. Stay tuned. We have many times on the podcast before talked about symbiotic relationships.
Mm-hmm.
We have, for a bunch of this episode already, talked about symbiotic relationships. One of the most famous examples of symbiosis, maybe the go-to example of symbiosis in the world, are lichens. We have talked almost nothing about lichens ever on the podcast because they just don't come up all that often in the kind of conversations we have. But this is an episode about plants and fungi. And not only were a bunch of the requests for this episode specifically requests about lichens, but now's the time.
Mm-hmm.
This is the time to do it. Allie, please tell us, what's the deal with lichens?
Oh man, that is such a good question. So, all right, I'm going to let y'all know how much of a big old plant nerd I am. Do y'all remember in 2016 when we found out what the third member of the lichens are?
I do remember when this happened.
Yes.
Okay, like I remember when this came, like when this news, which is also wild because like that's very recent, and also, oh man, how nerdy is that to get excited? Like, oh, we know, like we learned this thing about lichens. So to back up to explain that, lichens are a colony of green algae and or cyanobacteria living among filaments of fungi as well as a yeast in a mutualistic relationship. So we thought there were two, but there's normally at least three, if not more.
Yeah.
Which is wild. It is wild that all of these things are coexisting.
Yeah, this one is interesting because you look at like diagrams or microscope images of what a lichen actually looks like, and it's that fungal net with just individual cells sort of sitting in it.
Yeah.
That are the algae cells.
It is absolutely wild. And so it is unsurprising that we're still figuring out what on earth is going on with this. Mm-hmm. So there are roughly 20,000 known species. The estimates are close to 28,000. So we got a ways to go, but this isn't like a particularly speciose group.
Right. Now, I do have to stop you for a second because you've done a thing already that exposes the weirdness of lichens. Mm-hmm. So lichens is the word lichen refers to a structure.
Yes.
And the structure is the combination of the fungal mycelium and the algae or cyanobacteria and the yeast and whatever else is making up that structure. Who else
is in there?
And you just referred to them as species.
Yes. And that is something that you still commonly see because they're often the same sort of groupings. And so this is a whole thing that I get in a little bit is like what is an individual?
Right.
When you are made of 2 to 3 different kingdoms of life. Like, that's the wild thing. Also, side note for why herbaria are the best collections. We just casually have multiple kingdoms of life. In zoology, everything's an animal.
Yeah.
There's only one.
Everything that's supposed to be in there is an animal.
This is true. And so in the herbarium, we have plants, fungi, and whatever a slime mold is. So, like, just throwing that out there. We are the best. So I will say species because it's a useful analogy for talking about them, but it is not reflective of the reality. And as y'all mentioned in the last episode, we're so far away from what our animal minds understand that you kind of get into the danger of misrepresenting things by using these analogies. But, it is useful to a certain degree. So literally, that I would like to point out, David, the next point on my outline says, what is a species when the organism doesn't come from a single lineage?
I know how to do a podcast.
Literally a professional. And so, this is the question. This is part of the reason why it is so hard to deal with these things. So historically, we have dealt with these things in a variety of ways. Currently, the best practice, that's the thing that we're moving towards, for lichens is to classify them by the fungal component, so by the mycobiont. So if you're giving a scientific name to a lichen, it is the name of the fungi.
Okay.
So it's the name of the fungus, and then the alga has its own name.
Okay.
Right? So the lichen, which is the structure, right? The lichen is the fungus. Gotcha. And then also this algae is here.
Gotcha. All right.
Okay.
Right. Which, so this has only been a thing since 2014.
Wow.
10 years.
Yeah.
10 years. How,
It is sort of comparable to when we were talking about plants in the first half of the discussion, where you have a tree and the name of the tree is whatever the species of tree is, even though it is also inseparably intertwined with fungi in the roots and fungi within its tissues. We call that unit pine tree, pinus whatever.
Yes.
And then we call the fungal component whatever the fungal component is. So that is sort of similar to what you're describing.
It feels fair, honestly, right? Like, if the tree gets to have the name, let the fungus the structural component
gets to
have the name. Like, that checks out for me. So the tricky thing with this is that a particular species of fungus may form lichens with different species of algae. So the name that would appear, like, when classifying it is the same, right? Because it's a fungi name. But historically, the lichen species they were all given different names because they looked different, because they had different algae. It's complicated. And so this integration is something that's ongoing. We've only been doing this for the last decade or so. So before this, like, historically, lichens were actually placed in their own division. So thing to keep in mind when we're talking about plants is plants don't use phylum, they use division. So kingdom and then division is the next order or next group down, right? So the biggest umbrella term is kingdom. The next subdivision is division in plants, and also fungi, because fungi play by the taxonomy rules of plants. It's a whole thing. So previously, lichens were put in their own division, the Mycophycophyta, which is great because that name means fungus, algae, plant.
Yep.
It's so good. 2 of those things are correct. But that is no longer accepted because the fungal component can be from all over the fungi tree. The fungus in this relationship, when you're making a lichen, provides the structure, the moisture, and the nutrients, right? Which makes sense. This is pretty similar to the relationship that it had with, say, mycorrhizum, when it was a mycorrhizum, except for the plant was previously providing the structure. The photosymbiont, so that is the whatever flavor of algae or cyanobacteria, they provide the carbon, the carbohydrates via photosynthesis. The yeast, because don't forget, there's actually 2 fungi in there.
So, to remind people from last episode, yeast refers to, broadly speaking, any variety of single-celled fungus.
Yes, like you mentioned, it's like a tree.
Yeah, so you have this net of mycelium with individual cells of algae/bacteria and also individual cells of yeast.
Yes, exactly. So we have fungi, algae. It's very exciting. So the yeast also provides a little bit of shape when making the body of the lichen, the thallus. So it's found in the cortex, which I'll talk about right now. So the algae can live without the fungi. The fungi typically cannot live without the algae.
Okay.
Interesting.
So the term lichenized fungus is useless. It is used in it's utterly dependent on context because it is used in so many different ways. So lichenized fungi can refer to a lichen is a lichenized fungus, right? But also species of fungus can be lichenized because they form lichens. It's a whole thing. But in general, lichenized fungi, like the fungi that live in lichens, cannot live freely. They have to live with algae. Cyanobacteria, if you get one of those in there, so blue-green algae, they can also fix atmospheric nitrogen. Right? So, you can have an added bonus in terms of, someone who's bringing nutrients to the table. So the fungi, not the yeast, the fungi, the structural component, is typically either an ascomycete or a basidiomycete. Again, unsurprising. You can refer to the type of lichens that they make as ascolichens or basidiolichens. Makes sense. I love the obvious names. Approximately 20% of fungal species live as mycobionts.
Wow.
That is higher than I expected it to be.
Yeah, same. And approximately 40% of the ascomycetes.
Wow.
This is a very common life strategy for fungi.
Yes.
Holy cow.
Yeah. Which is also surprising given that there are, air quotes, 20,000 species of...
Right. Air quotes 20,000 air quotes species.
Yeah.
Yes,
of
exactly.
air quotes lichens.
Yes. Boku air quotes here. So as I mentioned, kind of the lead up, that yeast partner, and it's basidiomycete, that yeast, that has only been known since 2016. And suddenly it's like, oh, this makes sense. Because one of the big difficulties with studying lichens is you can't really grow them in a lab. And it's because they were missing part.
Yep.
Yeah.
Yep.
So if you don't have all of these components, you're not going to be able to grow one. So it's really cool. Lichens are more than the sum of their parts. They have different morphology, physiology, biochemistry together that each partner has alone.
Since last episode, I was waiting to use my underworld quote. Lichens, half fungus, half algae, stronger than both.
Yes.
But now
they are.
But now the yeast ruined that.
2/3 fungi.
Yeah. That quote just doesn't work.
2 parts fungus, 1 part algae.
Yes. Yes.
Mixed to taste.
Yeah. Yes, exactly. So there are 2 species in 2 different genera of green algae that are found in over 35% of all lichens.
Wow.
But they can only rarely be found living on their own outside of a lichen. So they can be free living, but clearly they choose not to.
Yeah.
So 20% of these fungi exist as lichens, but 1/3 of lichens are host to only 2 species of algae.
Yes.
So this is again like the mycorrhizae situation in reverse.
Yes.
That you have a diversity of structural component symbiotically existing with a smaller diversity of another symbiont that have been able to colonize essentially many different types of their hosts as it might be.
It is wild.
They're just breaking all the rules.
Yep.
Absolutely. Like, that is the thing about lichens, is it feels like it's the kind of the running joke is, everything makes sense until you add biology. But I
Sure.
feel like this is the prime example of, like, you thought you knew the rules.
Yeah. Well, I think it's so funny that we on the podcast, normal episodes of the podcast where we talk about cool animal stuff, Will and I are exploring, and every now and then we'll find an animal and we're like, that animal's weird.
Yes.
And then we do an Allie episode and Allie tells us about plants and we're like, that is alien. Like the way that plants function is just fundamentally different from animals. And now we're having an episode where we get to sit down with Allie and watch Allie go, these organisms don't make any sense.
Yep.
This is what it takes.
This is a fungi. We had to go to a different kingdom in order to freak Allie out.
I said it before, I'll say it again. Like fungi are so weird. And so you have this temptation to try to take a step forward. You don't need to make fungi any weirder than they already are.
Yeah.
Oh my god. I love that.
It's the dual danger of either the temptation to take their weirdness to the next logical conclusion and add unnecessary weirdness, or to try to make them understandable by using human and plant analogies
and misrepresent
that is now going to confuse you.
Yes, exactly.
They're in this narrow band of reasonable weirdness that you just kind of have to accept.
Well, and the thing about lichen too is that they look like they should be plants.
Yes.
Right? If you just look at that, you see it outside and if you told me that was a plant, I'd believe you. It doesn't help that they often have moss in the common name.
Yeah. It's very similar aesthetic.
Yes. So they have moss in the name. It seems like they should be plants. They actually just use plants as substrate.
Mm-hmm.
They don't parasitize them. They're just sitting on them. But that's not the only place that they will sit, not the only substrate they have. So they can be found in pretty much any terrestrial environment, from sea level all the way up to alpine regions, and some marine. There are marine algae that can form lichens with fungi.
That's awesome.
That upset me when I found out. That feels against the rules.
Yeah.
So in terms of substrate, right, you got your bark, your leaves. They can be on top of mosses. They can be on top of other lichens. You can have the epiphytes that are hanging from branches, just crusts on rocks. They can be soil crusts, just on top of bare soil. They can be endoliths, so inside rocks, and they can basically be between the grains of rocks. Sometimes they'll just pop up for their reproductive bits, which we'll talk about in a little bit. They could be on toxic slag heaps. Yeah,
well, this is part of that, as you said, more than the sum of their parts.
Mm-hmm.
Like, one of the things that is most famous about lichens is that working as a unit, they can live in places that basically no other organisms can survive.
Yes, absolutely. I love them. They're so weird. So they're often identified by their growth form and their color. I actually, when I was an undergrad and I went to the University of Michigan Biological Station, one of the things we did in my ecology class was learn to identify the different growth forms, the common growth forms of lichens. So the shape of the lichen is usually determined by the organization of the fungal filaments. So the fungus is what drives the shape of the lichen, which makes sense. It's the structure.
Yes.
So some of the common names of the common categories are pretty obvious. I'll say the name and you're going to go, I understand what that means. So there is, well, except for the first one I'm going to tell you. So fruticose is misleading. It doesn't look like it's fruit. It is three-dimensional. So they're tufts, or they almost look like little bushes, and have a more complicated structure. There's foliose, which looks like a leaf.
Mm-hmm.
Yeah.
They are two-dimensional. They have these leaf-like lobes. Then it's exactly what it says in the tin. And then there's crustose, which looks like a crust on a rock or whatever. And there's a bunch of others, but a couple that I thought were interesting and worth mentioning. Leprose, which are powdery looking.
Okay, interesting.
Right. And then gelatinous.
Yeah.
Cool.
Yep, they can be jelly-like.
There are jelly fungi. I don't know if
Yes.
that's a related group of fungi.
Yeah, I am not sure the specifics. I just like that lichens can look gelatinous.
Jelly lichens.
Jelly lichens. I love everything about it.
A gelatinous lichen.
So the like I said, the shape is driven by the fungus, but the color is usually determined by the photosynthetic component, which makes sense.
Yes.
Like, you know, we're using color for this. So in general, typically lichens are green when they're wet. So when they are wet, lichens can be bright green to olive gray. When they're dry, they tend to be grayish green, brown, something like that. If they have special pigments in them, so for example, ooznic acid, that's a yellow colored thing. So they can give them colors like reds, oranges, yellows, and browns, especially if they're growing in dry or exposed habitats. So if a cyanobacteria is in the mix, that's going to impact the color. Dry lichens that have cyanobacteria tend to be dark gray, brown, or black.
Yeah.
Okay.
In general, you kind of categorize lichens into two, I don't want to say tiers. They're not tiers because that implies a ranking. But, you know, two different umbrellas. So the macro lichens are the ones with the more complex structures. So your fruticose, your foliose, those sorts of things. And then micro lichens tend to be more uniform in shape. So macrolichens can have up to 3 different types of tissues, and these are differentiated by the density of the fungal filament. So this is kind of what we were talking about before. It's wild. If you look at the cross section of a foliose lichen, if a leaf-shaped lichen, if you look at the cross section of that, it looks like a leaf.
Cool. We talked about the leaf anatomy in episode 195.
Yes.
Where you revealed to us the tissue structure
Yes.
in leaves. I was wondering if you were about to say that.
Yeah, no, it is pretty much the same, and I don't know how I feel about it because it makes a whole lot of sense.
Yeah.
That's the shape that works.
That's the shape that works, exactly. So in leaves, you have basically like the epidermal sandwich, right? You have epidermis on the top and bottom, then you have so going from the top to the bottom, you have epidermis, and then the photosynthesis happens towards the top, and then you have the empty space towards the bottom, and then you have the bottom part of the sandwich. That's the same thing with these lichens. So you have these dense woven filaments, the cortex at the top and at the bottom of, again, this like photosynthetic sandwich. And then near the top of the sandwich, where I guess the lettuce would be, you have the photobiont layer. So that's where your little algae are hanging out. And then below that, you have this open space of the medulla. So the fact that it is functionally the same thing, genuinely, literally, functionally the same thing, is kind of wild. But you do have to keep in mind that lichens do not have stomata.
Yes.
Right. The gas exchange structures.
Right, exactly. And that will come up again in a little bit.
Well, they're also often on a substrate.
Yes.
Whereas a leaf, both the top and the bottom of the leaf are exposed to the air.
Yes.
As you'll remember from episode 195, when Allie blew my mind by revealing that the top and bottom of a leaf are different from each other. That a lichen doesn't have an exposed bottom much
Correct.
of the time. You're not exchanging gas through your substrate.
Right, exactly.
It is very fun to think the multiple fungi and algae coming together to make what plants grow as leaves.
Mm-hmm.
Yes.
And leaves are, look at what they have to do to achieve whatever the line is.
Yep.
Yes, exactly. Get on our level. So it's also, again, I'm going to say something and then I'm going to have to justify it because they're lichens. So they have extremely long lifespans and they are considered to be some of the oldest living organisms. But again, we're getting into what is an individual? Like, how do you really measure this?
Yeah.
So there are some specimens in the genus Rhizocarpon. So these are found in Alaska and northern Canada. So I taught 2 different species that are estimated to be approximately 10,000 years old.
Wow.
Nuh-uh.
Wow.
all I know is that is the kind of time span that just like that's the end of the ice age, right?
Yeah.
Those lichens saw mammoths.
Yeah.
Yes. That was my first thought. That was my first thought, right? When you're talking about like, there were pygmy elephants when the pyramids were built. There we go. Yeah, same sort of thing. Like there were mammoths when these when these lichens were born. Again, it's a whole thing. But speaking of where baby lichens come from,
Yes.
unsurprisingly, they can reproduce vegetatively, right? You break off a chunk and there you go, you got a new lichen. Or they can send out diaspores. So basically these are lichen propagules that contain a few algal cells surrounded by fungal cells. And you can basically seed new individuals in air quotes. Basically assume that I'm just constantly air quoting when I'm talking about things.
These spores feel like more like escape pods
Yes.
than a true spore.
Well, it feels like a seed.
Mm-hmm.
Yes.
It
yes.
has the plant component on the inside and it's surrounded by a protective layer.
Yes.
But in this case, the protective layer is going to then grow into the new lichen structure.
Yep.
Yes, exactly. But this is the thing that I don't know why this didn't feel allowed, but they can reproduce sexually.
Yeah.
And that,
No.
David's reaction, right?
I had heard that before, and I had that same thing of like, sure, I guess, fine. Mm-hmm.
Okay, so let me explain what that means, because it's not. When I read, I had to take a lap. I needed a moment because it made me question everything I know about biology. So in general, sexual reproduction is selected against in the photobiont, mostly. So in general, the photobionts tend not to reproduce sexually. But lichenized fungi, so fungi that live in lichens, they can reproduce sexually, like a normal fungus. And just send out their spores. But these spores must meet a compatible algal partner in order to form a lichen.
Okay. It's a gambling shotgun shot of you're hoping you land near algae.
Yes.
So the asexual reproduction version can be we bundle up some fungus and algae together and we send it off as a collective spore to go grow into a new lichen.
Correct.
But the sexual reproduction version is just the fungus reproduces and sends out its spores. And if you meet an algae, you get to be a lichen.
Yes. Then you get to be a throuple again.
That's phenomenal.
Isn't it? It also makes me feel a little bit better about the whole process.
Well, and we talked about last episode that fungi can create a lot of spores.
Yep.
Oh, yeah.
So you're playing the numbers game.
So, in short, oh my god, lichens, they're wild. But, funnily enough, some fungi are actually parasitic on lichens.
That makes all the sense.
Yeah.
Which I just think is so funny, because lichens are 2/3 fungi.
Yep.
Well, and it sounds like you've got moss and then a lichen grows on the moss, and then another lichen grows on the lichen.
Yep.
And then a parasitic fungus moves in and parasitizes that lichen. And next door, there are some ghost pipes that are plants that are parasitizing fungi that are in a symbiotic relationship with the roots of a tree, where higher up in the tree, there's a bunch of epiphytes that are hitchhiking, and sometimes they're pathogens.
Yeah.
All of it, these episodes talking about plants and fungi really does drive home the myth of individual organisms.
Yes.
Oh, absolutely.
And like we animals are not exempt from this by any means.
Yeah.
But it becomes so apparent when we start really getting into these relationships.
Absolutely. Absolutely. And the thing about it is, like, lichens are so common, and there are so many things that are lichens that you don't realize are lichens. So some of the most famous lichens are like reindeer moss or reindeer lichen that's found in Scandinavia. But Spanish moss is a lichen.
Yep.
Really?
Yeah.
Oh,
Yeah.
cool.
Isn't that awesome?
Yeah.
That's my favorite.
Yes.
That is by far my favorite. I grew up in the South. That is my
Yes.
favorite lichen.
I have friends who are doing field work in Florida right now, and so I've been sent so many pictures of Spanish moss, and I don't have the heart to say that's not a plant, but I love it. It's my favorite lichen, so like I'm not complaining.
It's the best.
Oh, they're so cute. So, all right. There's a lot of drama when it comes to lichens and the evolution and like the fossil record of lichens. You don't know the half of it.
I bet.
I don't know any of it.
Okay, I'm gonna give I'm not gonna get into the tea, just a brief overview, but unsurprisingly, we were talking about how all of the fungi fossils, the paleobotanists have them. For it's kind of a similar thing where when people, non-paleobotanists, find material, it can be confused as lichens. The number of people who find weirdness in the fossil record and then oh, that's a lichen. The ones that disappoint me the most is when people who really should know better find dendrites, which is basically chemical staining on rocks.
Yeah, that's just minerals. That's not even alive.
That's magnesium. And say that they're lichens. So there has been if you dig into the literature about the fossil history of lichens, you'll find all this evidence from the Paleozoic, this and this and this and this and this. And there's a paper that came out in 2014 that basically went through as there is no evidence to suggest that lichens evolved before land plants.
Mm-hmm.
Interesting.
Yes.
Gotcha.
So because that was the thought for a very long time, and it was based on a lot of really shaky evidence.
Yeah.
And that's the thing. We were kind of joking earlier that these aren't charismatic fossils. They just they're squiggles on rocks, and it's true. They are variable squiggles on rocks, and so it can be really difficult, especially when you're dealing with a species like lichens, right? Being able to parse that and really figure out am I looking at some sort of early plant, or am I looking at some, I don't know, biofilm, or is it a lichen? And so there is no evidence to suggest that lichens evolved before land plants.
Okay.
But hey guys, it's your turn to guess. How many times did fungi that can form lichens evolve?
What did you say earlier? That there were 20,000?
So there's 20,000 species.
species.
Yeah.
Yes.
All right,
keep
there we
in
go.
mind,
28,000 times.
There we go. There we go.
Keep in mind, it's both basidiomycetes and ascomycetes.
Yeah.
So
I mean, it's got to be 1,000 times. This has to have happened a bajillion times.
Okay, what's your final answer?
A bajillion.
Bajillion.
That's our guess.
Big B.
5.
Allie, beautifully done. That was very well done. 5?
5?
Listen.
That's only slightly more than there are components of a lichen.
As a person who does not have any expertise in plants or fungi or lichens and has not engaged with any of the research that went into this, I choose not to believe that.
Yeah, no. Once again, I disagree.
Yes, I did. Surely they're wrong.
It's wild, right? I have been lulling you into this. Oh, it keeps happening over and over and over. So it seems like possibly 5 times, 3 times in basidiomycetes, and maybe only twice in ascomycetes.
Wow.
So that makes it, whereas the mycorrhizae, and every time we talk about parasitism and all that stuff, we talk about how this is such a successful life strategy that it happens over and over and over and over again, right? Fungi living around plant roots and helping with nutrition and all that is such a good idea. It is so effective for survival that it has developed over and over and over and over again. This makes lichens feel more like something like flight.
Yes, exactly.
Oh, yes.
That it only has actually evolved a small number of times, but it's such a beneficial, such a successful living strategy that when it evolves, it explodes and it
Yes.
gives rise to a huge diversity of descendants.
Which is a very cool parallel just from like a lessons on the nature of evolution, that you can get very similar pictures with very different ways. One is numerous because everyone's doing it. The other one's numerous because it was super successful when a few people did it.
Because
Yes.
everyone is descended from the first few that did it.
Yes.
Exactly.
Yes. No, it's wild. Like my favorite thing is to talk about how many times things arose, because I love watching y'all guess.
Yeah.
And the fact that I got you so hyped up that it's going to be a big number. Ah, delightful. No regrets. So one of the cool things about lichens is they are pioneer species.
Yes.
Right? They tend to be some of the first to show up in places. They can live in truly inhospitable places. And one of the reasons for that is because they are poikilohydric, which means that they can completely dry out.
Yeah.
You had mentioned that before, the difference between dry and wet lichens.
Yep.
Yes, exactly. And so the fact that they can dry out, just like this is my life for a while, and then rehydrate and come back, could be one of the reasons, why they're so good as pioneers. But also, like, if you're going to live for 10,000 years, you need a break sometimes.
These two things were the first things I learned that made me a fan of lichens, because I learned about them being pioneer species, and the documentary I was watching was like, very likely if we ever in the far, far, far sci-fi future start like terraforming planets, lichens may be a tool in that, to be some of the first life forms we put on a barren rock that we are wanting to create an ecosystem and environment in.
Yeah.
But also, when I first got into model making, my dad introduced me to using lichens for that, and because it makes good little bushes for a scale mini. But he was like, "Yeah, if you put that somewhere with water, it'll start growing again." And I was like, "Really?" He's like, "Yeah, and it'll just come back."
That's cool.
Yeah, so I had a whole bunch of lichens in my thing for my minis that were all dried out, but I was just always like, "You're still alive."
This also has echoes of episode 199 when we talked about hibernation
Yep.
and
Yes.
torpor and dormancy.
Mhm.
Very cool.
It's very,
Yes.
very tardigrade of them.
Yes. So, Allie, I'm going to ask you another question that I already know the answer to. Are there fossil lichens?
I'm going to ask you a question I don't know the answer to. Are there fossil lichens?
Yeah, but not a whole lot.
That makes a lot of sense.
Yes. So they have a not so great fossil record because they are common in places that are not well represented in the fossil record.
Yeah.
Oh, yeah.
Right? So they're common in the tundra, in the mountains, and in the desert. So they're not set up for success in terms of making it into the record. But they're there. They are there. Unsurprisingly, we found them in Baltic amber.
Sure.
Yeah.
Because amber is a great place to find these sorts of things.
Especially things that grow on trees.
Yep.
Yeah, exactly. It was in the right place. So Baltic amber is from Europe, Eocene, roughly 40 million years old. You will also sometimes find lichen fragments with leaf fossils.
Okay.
So again, if you can get some really exceptional preservation, you can tell the difference. There's some known from the Miocene of California. Again, these are much more recent things, which makes a whole lot of sense. So there are the oldest fossil lichen, and this is one of the things that's like, I don't know how much I buy this, but the oldest fossil lichen is from the Rhynie Chert.
Sure. That's the place that it would be from.
Now, is this lichen still alive? Because so far, I just feel like I need to check.
This lichen was best friends with a trilobite.
Yeah, right. Like, is this one still around? Because so far, I don't know what to expect with any of these.
So I don't think they live 400 change million years. But as I mentioned, the record for the evidence of Paleozoic lichens is really contentious because it can be hard to figure out what is a plant, what is a lichen, I have no idea.
Yeah,
what
some other weird thing.
Yes,
Yep.
exactly. So in terms of like actual evidence, it's way better as we get closer, but in general, it's not great based on where they live. And also, if you think about what they look like, if you have a powdery lichen, how well is that actually going to show up in the fossil record?
Well, like gelatinous ones. Like,
Yes.
can that even preserve?
Right, exactly. And so the fruticose, the foliose, these macrolichens are way more likely to show up in the fossil record just because, like, they're big.
Mm-hmm.
You have, more opportunities to be seen.
Yeah,
Cool.
this is a theme that's sort of evolving. We talked in the last episode about how the early fossil record of fungi in general is pretty sparse, that there are some maybe Proterozoic fungus
Mhm.
fossils, but they're not really known until the early Paleozoic. But also a theme that is especially prominent when in this discussion about lichens is that our understanding of the fossil record and evolutionary history of fungi and their relationships, the biggest limiting factor to our understanding of that is our limited understanding of modern fungi.
Yes,
That, like,
absolutely.
we don't, and this is something that is easy to say sort of lightly, but most of what we understand about the deep past and life in the past is based on what we understand about life today.
Mhm.
Like, if you don't understand plants, you can't interpret fossil plants.
Yes.
Right.
And so as our knowledge of modern organisms improves, it feeds into our knowledge of the past. And if the lichens are still such a interesting bundle of mysteries for us today, it makes total sense that we don't have a lot of insight yet into their history.
Yeah.
Study lichens more.
Yes.
The other thing that when you were talking about them being super, super old and all that, it made me think of when we talk about coral reefs.
Mm-hmm.
And you will have that where we will describe this reef system is X number of thousands of years old.
Mhm.
And it's not necessarily that it's the same coral, but it's building
Right.
up, and you get this sort of buildup. And I had that when you described the lichens being ancient, I had that same thought of, is this sort of a coral reef
Right.
thing going on?
Well, it's got the same thing as talking about like the age of a city.
Right.
Where it's like a lot of these buildings aren't the same buildings that the city was founded with. They've been rebuilt and they've been, changed. I thought about coral a couple of times because also of the algae being able to survive on it. Like the zooxanthellae coral also are many corals are dependent on them.
Mm-hmm.
The algae can be in the water anyway,
Right.
by itself.
Exactly.
So another major theme over the course of this episode is how important plants and fungi are to each other.
Yes.
Right? That in the world today, you basically can't have plants or fungi as we know them without them being sort of linked together. I know that it is a major question of how deep do these relationships go? How intertwined is the coevolution between these groups? And how significant has it been to the development of plants and fungi as we know them?
I'm going to talk about terrestrialization.
Excellent.
To begin with, I have a couple of other things I want to cover, but terrestrialization, I think, is the most important aspect of this relationship between plants and fungi, because mycorrhizae helped lead to the terrestrialization of plants.
Yeah, that's what I was wondering.
Mm-hmm.
Basically, would not have plants in the way we have them now on land without mycorrhizae. So they helped the plants acquire water and nutrients. And this, keep in mind, this is before the evolution of extensive roots. And so if you can get a buddy who basically functions as roots, that can help you move further away from the water.
Yeah. Will, earlier you joked about it being the buddy system.
Yep.
But yeah, it might actually have been that the plants and fungi had to move onto land together.
Yep.
Yeah. So mycorrhizae are possibly as old as the terrestrialization of plants. Like mycorrhizae might date back to plants coming onto land like 470 million years ago.
which is extra fascinating because it would suggest, and I don't know if we have enough detail to really say for sure anything about this, but the fact that mycorrhizae are so ubiquitous, so widespread across modern land plants, A, makes a ton of sense because it's probably ancestral.
Mm-hmm.
But also it means that stuff like brassica that doesn't have mycorrhizal fungi, it is more likely that they lost their mycorrhizae than that they never developed them.
Right, exactly. So the molecular clock is what a lot of this is based on, again, because like trying to really parse, like especially these earliest fossils is
Sure. So
rough.
this is based on genetic evidence.
Yes, and pinning things with fossils whenever you can. So as I mentioned before, the arbuscular mycorrhizae came first. They are the OG. They are plants' buddies from the beginning. So basically, the evidence suggests that they arose about the time that plants came onto land, roughly 470 million years ago, and were probably a driving force to allow that to happen. Ectomycorrhizal fungi didn't arise until the Jurassic.
Around the time that those angiosperms were getting their start.
Which makes sense, right? Because they have this deep association with angiosperms. So it's interesting to try to figure out why. Why do we have this new type of mycorrhizae, right? Why do you have one that is not integrating itself into cell walls, instead is making the sheath structure? And it could be for a variety of reasons. So some of the ideas that it could be because of changes in climate. So increased seasonality and increased aridity, because I mentioned that the arbuscular mycorrhizae are not really found in more arid environments. And it could be in response to more nutritionally deficient habitats. So all the other types of mycorrhizae that I didn't talk about pretty much arose in the Cretaceous.
Okay.
With the radiation of angiosperms.
Interesting. So plants started doing something different.
Yes.
And hit it big.
Yep.
And it seems like fungi either hitched a ride along that, right? They were able to diversify, or were an important component of allowing that to happen in some way.
Yeah.
Yes, exactly. So whether or not it's one or the other, it's probably both, because it's biology. It is always both. So that's terrestrialization. We couldn't have done it without fungi. Again, I am a plant here.
The fact that they may have been so critical to terrestrialization also makes plants and mycorrhizae feel like a pioneer structure, like lichen. Like
yes.
That's
the OG lichen.
Exactly. That feels very similar.
Yes.
In regards to, it's just lichen, but the roles are switched.
Well, and it kind of drives home the comparisons that we were making before, that we look at lichens and we go, wow, what a weird group of organisms that are multiple. It's plants plus fungi existing as sort of a unit, and that's why they're able to live where they do and how they do. And the reality is that that statement is kind of true of all land plants.
Yeah.
Yes.
Which is wild.
Yep.
Yes. We talk about, no one pays attention to plants. No one pays attention to fungi even more.
Yeah,
exactly.
But they're besties. They're in it together.
Cool.
So that's terrestrialization. So I'm going to kind of pivot a little bit and talk to you about some things that have to do with, human influence. Invasiveness. The relationship of fungi to plants can play a role in how invasive a species can be.
Oh.
So plants that are not native to an area often require their native mycorrhizal symbionts in order to survive. So the vast majority of arbuscular mycorrhizae are non-specific. So the plants that interact with these mycorrhizae can become invasive very easily, because it doesn't matter who it is, as long as it's somebody. If you have, so the ectomycorrhizae tend to be more specific. So if you are trying to introduce a species to a new landscape, you may need to introduce its native mycosymbiont. So for example, if you're talking about exotic forest plantations, So, like, specific examples of this are pines in the southern hemisphere and eucalyptus in the northern hemisphere. So they are both obligate. They have to have their ectomycorrhizae in order to survive. So in their native range. So pines were difficult to establish in the southern hemisphere until they inoculated the forest with their mycorrhizae. Same thing. So eucalyptus plantations in the northern hemisphere require this fungal inoculation in order to grow.
Yeah.
Fascinating. Also, a little bit terrifying.
Yeah.
Yes.
Because it's like saying, in order to move this species of animal into this new habitat, we also have to bring all of their bacteria with them.
Yeah.
Yes.
Which is true. I
Yes.
mean,
Well,
that
yes.
is also
Exactly.
true.
Yeah, I was thinking the same thing of it's that issue of you can't just take a plant seed necessarily and take it anywhere and start growing it. Just like you can't just take a baby animal and it be good to go if
Right.
it hasn't had a chance to get its gut bacteria.
Yes.
But this one has the even further step of being weird and creepy in that, like, to transplant a plant from somewhere else, you might have to terraform the soil
Yeah.
with their mycorrhizae.
Well, and you see, you had to inoculate.
Yeah.
That's crazy.
That feels more like if you want to move this animal to this place, you have to bring the air that it needs.
Yep.
Yes.
It's like aliens need to bring their methane tanks or whatever to the planets they're going to.
Exactly. And we had kind of talked a little bit about plant competition, right? So, have, ways that plants are, they have crown shyness, so they're not overlapping with each other, or they'll try to crowd each other out, all of these things. Another way that plants can compete is by attacking each other's fungal networks.
Yeah.
That makes sense.
Yeah. You can use your powers for good, or you can use them for ill.
Well, you can cut off the supply line.
It's cutting the power lines. to the building.
Yeah.
Yes, exactly. So dominant native plants can inhibit the ectomycorrhizae on the roots of neighboring plants, and some invasive plants can inhibit the growth of the native ectomycorrhizal fungi. So that's a way that they can become invasive, a way that they can become dominant. A really good example of this is garlic mustard. So garlic mustard will release a chemical that inhibits the growth of the ectomycorrhizae that live on pine seedlings.
Oh.
So, like, basically they're excluding their competition in that way.
Yeah. That's so cool.
So building on humans, doing things to the planet, inoculating the soil, etc. So talking about climate change and the human impact of these relationships between plants and fungi. So it is very unclear how climate change impacts mycorrhizae, because there are studies that say it makes things better. There are things that say it makes it worse. There are things that say there's no change. In general, the mycorrhizae tend to do better in cooler temperatures. But most biological processes tend to do better at cooler temperatures. In addition to that, fertilizer has a massive impact on plants' relationship to their mycorrhizae, which makes sense because the mycorrhizae's job is to bring in these nutrients. So if humans add fertilizer with a bunch of nitrogen or phosphorus, it can lead to plants ditching their fungal symbionts.
I was wondering, because you mentioned in more nutrient-rich soils, they don't often have it because they don't need the help.
Yes. So, and here's an example in Alaska. So there was a study that looked at 30 species of, that looked at these localities across Alaska. So in the low nitrogen sites, you would get 30 species of fungi in the soil. And at the high nitrogen sites, you would get 9.
Wow.
This is really important because remember what I said about the ectomycorrhizae. They cannot complete their life cycle without a plant host, which means that, like, if they get ditched by the plant, that's it.
Yep.
Yeah. They go extinct. They're gone.
Right. And this changes the fungal makeup of the soil.
Yeah.
You are like, this is a big deal.
So like, if you fertilized a place for a couple of years, you could end up wiping out the majority of your mycorrhizae populations.
And
Yes.
and not only that, you could end up with a population of plants that now can't survive without your
that
fertilizer.
if you suddenly stop fertilizing, they now no longer have their helping hands.
And that's not always enough. So I mentioned phosphorus. So if you add phosphorus to the soil, then the mycorrhizae, like I told you, right? The mycorrhizae are 6 times better at bringing in phosphorus than your normal plant roots. If you add phosphorus to the soil, the plants would be like, cool, I don't need this, it's more energy for me to give to you. I'm able to get enough phosphorus on my own. But this can lead to deficiencies in other micronutrients because even though your fertilizer is giving you all this nitrogen, is giving you all this phosphorus, it doesn't have things like copper.
Yeah.
Right? So you have these other deficiencies that would normally be fulfilled by the mycorrhizae, but because we're just dumping all of these, dominant, normally limiting factors, we're running into different problems.
So you can give them enough phosphorus for them to ditch their fungal partner, but that now they are going to struggle to get all the other secondary things.
Yep.
That's very that feels very much like how I've seen the survival guides where they warn against, like, don't just eat rabbits because you will go into I can't remember, there's a term, but you're not getting enough fat.
Yes.
Because that's a lean animal. You will go into shock even if you are eating 3 full meals a day. But if it's just lean animals, it's got that sort of thing that you could accidentally hurt yourself by getting too much of one thing.
Right, exactly.
Not because it's poisoning you, because you're not getting a balanced meal.
So one last thing, kind of talking about climate change and human impact and all of that, and these symbioses. Lichens. Lichens are, like plants. They can't move around.
Yep.
Which makes them excellent environmental indicators, especially because they're photosynthesizing, right? So because they don't have stomata, right? They don't have these holes in them to allow for gas exchange. Basically, it's the whole surface of the lichen that is photosynthesizing, right? And is basically means that the whole surface of the lichen reflects the elemental levels of the atmosphere, right? Of the air quality, right? So different species have different sensitivities to this, but in general, you can use lichens to measure air quality. And do you know how long people have been doing this? How long have people been using lichens to monitor air quality?
Oh, I bet a long time. I have no.
Is this one of those back to the ancient Greeks?
Not quite that long.
Okay,
right. That's where my brain was going. It's like, were the Egyptians doing it? Because I wouldn't put it past them.
Yep.
So there's basically been a process for this. It was first documented in 1859.
Wow.
We barely knew what they were then.
Yeah. That's very awesome.
Allie, I think that this is the most fascinated that I've been in any of the episodes we've had you here on.
Mm-hmm.
This is barely about plants.
Maybe that's why. Maybe that's
the
it.
secret.
That's the trick. The trick is stop talking about plants. This is so cool. I'm very happy that we had you come in to do the fungi and plants episode, because you bring with you that plant foundation that really helps to drive a lot of these concepts home. Man, this is so. I thought this episode was going to be about 2 different groups of life, and not about the fact that those 2 different groups of life are not actually as distinguishable as we imagine that they are.
Yeah. Well, it also, and you made this point earlier, Allie, that people overlook plants. We treat them like they're the green background, and then we don't even overlook the fungi that are so closely tied to them because we are just fully unaware of them so often.
Yes.
To the point of not realizing that you may be sabotaging the soil of this area by over fertilizing, that in years to come it can't grow the plants it once could because you got rid of its allies, or you need to bring their allies places if you want these plants to grow. These invisible key elements to the plant that you're so familiar with. Very cool.
Allie, I'm going to ask you one last question. It is a question that I've asked on several of your episodes because I love asking this question about plants. How did fungi and plants together change the world?
I think it is not an exaggeration to say that the world would be fundamentally different if plants and fungi hadn't become besties 460 million years ago. Because it started the moment that plants came onto land. Because without fungi, we would not have terrestrialization of plants in the way we do. Like, I cannot even imagine how that would have worked without the help of fungi. So, right, if we have a different situation of plants coming onto land is what led to animals coming onto land.
Mm-hmm.
So I can't even, it's one of those things that this is just so, this was step one in many ways.
Yeah.
And so the fact that, we would have had this fundamental difference at step one means that, like, I can't even imagine the world that we would live in. How did they change the world? They made the world.
Yeah.
Right. Like, this wouldn't be here. We would not be having this conversation. Like, that's pretty much it. And like we've said multiple times, plants would not be so dominant without their fungal partners, but plants still get all of the attention, which is a weird thing to say because plants don't get any attention.
Yep.
And it's so funny because it's one of those things, right? I go, like, as a botanist, I go off and how no one, like, plant blindness and no one notices and da-da-da-da-da. And then the fungi collection manager is like, I'm gonna stop you right there.
Yeah.
It's just
somewhere out there in the world, there are 3 researchers who specialize in archaea, just shaking their heads.
Yep. It's that meme of the kids in the pool, and it's the animals being held up by the parent, plants drowning, and fungi, the skeleton at the bottom.
I have literally seen that meme.
Yep.
Allie, this has been so much fun. This has been exceptionally interesting. We have covered the bare surface of fungi-plant relationships, but it has been a super exciting discussion. We are going to stop because we have to stop at some point. Before we end the episode, we have one thing to do, and that is our patron question.
Yeah.
The podcast is supported by our followers on Patreon, and among the many goodies that you can get as a patron is the ability to submit questions for us to ask here on the podcast. This episode's patron question came from 2 different people who asked very similar questions that I thought was very fitting for this episode. Luke and Lori both asked, are there any animals who have developed photosynthesis? And if not, why not?
Okay, I am so excited about this. The answer is yes, kinda. There are multiple types of invertebrates that have some type of photosynthesis that they can rely on. They're not doing it themselves, though. So sea slugs are really famous for this. They will steal chloroplast, called kleptoplasty, from the algae that it eats. It basically incorporates the algae into its gut, and then be able to get the energy from it that way. But it is not passed vertically, right? So this is not something that is passed from parent to offspring. You have to do this on your own in order to get this. Earlier in the episode, Will, you mentioned zooxanthellae.
Yeah.
So corals, the giant clam, and a host of other things are hosts to this zooxanthellae, which are colonies of photosynthetic algae. So the animals are the structure that give the home to the algae, and the algae provides the animal with energy. It is variable whether or not this is passed vertically, whether or not the algae are passed from the parent to the offspring. It depends on the group.
Yeah.
The only known solar vertebrate is the spotted salamander, Ambystoma maculatum. Love them. They're adorable. So there is a species of algae that is found in the embryos of these salamanders. If you look at their eggs, they are green. It is great. They are in Chlorococcum ambistomatis. So basically, they are green balls, which like checks out. From they're green balls, and they are lovers of salamanders. So they're probably only found, well, the species is only found in embryos. Whether or not it gets passed on and continues to live throughout the life cycle of the organism is unclear. There is evidence, however, to suggest that it is passed from parent to offspring.
Okay.
So that's the closest. But a salamander is not very big.
Yes.
You are definitely size limited.
Mm-hmm.
And you either have to live in the water. Well, actually, that's it. You need to live in the water.
Yep.
So unless you live in the water, because it's algae that's doing this.
Mm-hmm.
So if you need to live in the water, adjacent to the water, have part of your life cycle in the water, you need to acquire an alga of some kind. Like, those are your limitations. The animals don't really... We've gone too far down the path to really become photoautotrophs. But there was a study that came out a handful of years ago that basically was like, what would it take
Yeah.
to have to power a human body with photosynthesis? This is my favorite fun fact. I have been rattling this off for years. So humans are big, right? We are large animals relatively, right? And so our demand for glucose is way higher than photosynthesis could accommodate. So in order for an adult woman, so for me, if I were to photosynthesize like a leaf and make all of my own energy, I would need to have a body with the surface, with the skin that is green, with the surface area of a tennis court.
Yep.
Oh my goodness. Like, just so if my body were just green, that would get me 1% of my daily need to a tennis court.
We're just too energy consumptive
Absolutely.
as organisms.
Our brains are eating a lot of sugar.
Yep.
Yeah. So animals have achieved photosynthesis sort of in the same way that fungi have achieved photosynthesis, which is either building a structure that is composed of you with some algae that are photosynthesizing, or eating plants.
Yeah.
Yes.
And getting the photosynthetic energy that way.
Yeah.
Yes, exactly. But yes, thank you for letting me share my favorite fun fact. I bring it up in conversation more than is probably socially acceptable.
I love that. I also wanted to throw out my favorite example of photosynthetic animal is upside down jellyfish.
Oh.
That their little bell heads, they swim downward, and their tentacles have photosynthetic algae in them, so they look like seagrass.
I love that.
And so it's camouflage and photosynthesizing, and then if you disturb them, they flip over and will swim away, and if they flip over on you, they'll sting you. And that's how my dad found out that it wasn't seagrass.
Thank you, Luke and Lori, for asking that patron question. Thank you, Allie, for coming on and talking to us about fungi and plants. This was super fun.
Learned so much.
I loved watching you be shocked by the words that came out of my mouth.
Listeners, go and check out the blog post with this episode on our website, linked down in the episode description. We will have some links to more information. We will also have pictures of a bunch of the stuff that we've been talking about. This has been episode 2 of our fungus trilogy that we are in the midst of. Next episode will be even more fungus discussions.
Yeah.
So if you're having fun so far, definitely come back for the next and final one, episode 200, part 3, episode 202. Fungi, and more fungi. Don't forget that our Spotlight series is ongoing this year. There will be another episode coming out at the start of October, shortly after this episode drops. Also, this episode comes out right before the start of October, and that's spooky season. So we will be releasing several episodes of Spooky throughout October. We've got Cutie in November and then a Spooky live stream. Join us for all that stuff.
Yes.
A huge thanks, as always, to all of the requesters, the many, many requesters who suggested that we do not only fungi in general, but also this particular episode topic. A huge thanks to all of our patrons for supporting us, for allowing us to do our science communication projects, for allowing us to get to episode 200 and beyond of the podcast. We are extraordinarily grateful. A special extra thanks to our top tier patrons, Danielle the Bug Lover, Melissa Barksdale, Robert Mart, and Sarah May. Thank you for your incredible support. And an enormous thanks, as always, to our favorite paleobotanist, Dr. Allie Baumgartner. Allie, thank you so much. This was great.
I'm so glad y'all let me do this.
Now people are going to want you to come onto their podcast and talk about fungi.
Yep.
I am not an expert on fungi, but I know some people who are.
There we go. See, this is the train that we've been building when people want to talk about stuff with us. We release episodes every fortnight. Listeners, join us for the next episode where we will finish out our big fungus extravaganza, at least for the time being.
Yeah, everything that'll need to be said about fungus will have been said.
That'll be it.
We'll have covered it entirely.
No more needs, unless maybe you'll find another place to go get your fungus information. Also, come back in a few fortnights, episode 205. Allie will be back because we got plant episodes to do, and those are the fives. I am out of things to say.
Yep.
Your homework for this episode is to go outside into the woods and stand there and ponder how many organisms you can't see.
Yeah, just stare at the ground and picture.
What are the trees saying to each other about you? Bye, everybody.
Bye.
Bye.
Thanks for listening to the Common Descent podcast. You can follow us on Facebook, Twitter, YouTube, and check our WordPress blog for pictures and links after each episode. Huge thanks to our patrons whose support helps keep this podcast running and who get access to bonus goodies on Patreon. The song you're hearing is called On the Origin of Species by Protodome, which we found at ocremix.org. Thanks again for listening. We hope you'll join us next time.